Hi, I'm Jim McAleer, the President and CEO at Alzheimer's Orange County, and welcome to the 32nd Annual Alzheimer's Research Conference, Alzheimer's from All Angles. As you probably know, Alzheimer's Orange County is an organization that provides care, comfort, and support to people who experience memory loss. But we know firsthand from those we serve how important research is and is going to be to all of our futures. We heartily support the conference and are very grateful for our collaboration with Dr. Joshua Grill and UCI Mind. I'd like to particularly thank all the staff of both organizations who spent countless hours to bring you remarkable, educational, and interesting content. Thank you, Josh. Thank you, UCI Mind. And thank you to all the staff and to you for coming and joining us. In just a few moments, you'll hear a little bit more about our work and what we do to help people experiencing memory loss. I hope you enjoy the conference. I hope you reach out if you're experiencing memory loss and we can help. Once again, welcome to the 32nd Annual Alzheimer's Research Conference, and have a great day.
and welcome to the 2021 Southern California Alzheimer's Disease Research Conference, brought to you by Alzheimer's Orange County and the UC Irvine Institute for Memory Impairments and Neurological Disorders, UCI Mind. My name is Josh Grill. I'm a faculty member in the Department of Psychiatry and Human Behavior in the UCI School of Medicine and in the Department of Neurobiology and Behavior in the UCI School of Biological Sciences, and I'm the director of UCI Mind. We are delighted to once again partner with our friends at Alzheimer's Orange County to bring you this year's Southern California Alzheimer's Disease Research Conference, and we're delighted that you've chosen to join us for today's exciting events. Of course, we had hoped that we would be able to return to an in-person venue for this year's event, but the lingering COVID-19 pandemic and our deepest commitment to the safety of our professional colleagues, our participants in research, our advocates, and our larger community dictated that we once again turn to virtual channels to produce today's conference. Though not our preference, there is a clear silver lining to doing this conference through virtual channels. This year, we have broken last year's record as it relates to registrations, with more than 1,250 people registered for today's exciting event. The last year has been a consistently challenging one for us all. Yet, UCI Mind and the field of Alzheimer's disease research have seen important and exciting progress and advances. In the field, the unparalleled event that occurred was the accelerated approval by the Food and Drug Administration of aducanumab, the first new treatment approved for Alzheimer's disease in 18 years. Aducanumab is a monoclonal antibody against the beta amyloid protein that accumulates in the brain of a person with Alzheimer's disease. This infused therapy has been convincingly shown to reduce the amount of beta amyloid in the brain, and it is this effect upon which the FDA based their decision to grant accelerated approval. This was not without controversy. The evidence that aducanumab slows the course of Alzheimer's disease is mixed, and the FDA has required the maker of aducanumab to conduct an additional clinical trial to determine whether aducanumab has clinical benefit for patients who are treated with it. Since this approval, 2 other drugs that have also been convincingly shown to lower brain amyloid levels in people with Alzheimer's disease have been granted breakthrough status by the US FDA. We are all awaiting news as it relates to potential added approvals for drugs to treat Alzheimer's disease, as well as key decisions around the use of aducanumab, most notably including whether Medicare will cover the treatment with this new drug. We at UCI Mind have been part of studies of these agents, and we've led important conversations about these controversial decisions and their potential implications to patients, families, and future research. We've also tried to carefully outline these events for you, our community, on our UCI Mind website. I'd encourage any of you for whom this news may be new to visit mind.uci.edu and read our timeline of events as it relates to the development and eventual approval of aducanumab. And I'd recommend any of you who have not yet done so to sign up to receive weekly blog notifications from UCI Mind. This year, UCI Mind also launched UCI Mindcast, a new multimedia platform offered through our website that now also features 6 additional different types of video, live stream, and podcast content posted regularly on mind.uci.edu. This includes continuation of our popular Facebook Live Ask the Doc series, as well as new content, among which one of my personal favorites is our Spotlight on Care, hosted by our dear friends and supporters, Virginia Nave and Steve O'Leary. Guests provide pearls based on their own experiences as caregivers, and Steve and Virginia are nothing short of masterful in guiding these important and informative conversations. This year, UCI Mind also started new educational programs. This includes the new Beal Scholar Program, supported by Don and Joan Beal, that provided comprehensive and, we hope, inspirational training to 16 local Orange County high school students who applied and were selected to participate. With support from HFC and Dr. Lorna Carlin, we began the UCI Mind RAMP program, matching UCI medical students for mentored research projects in the area of Alzheimer's disease and related dementias. Our Alzheimer's Disease Research Center research education component, led by Dr. Liz Head, took on a cohort of 6 early-stage investigators and faculty members to help them through the most challenging period of academic faculty positions, achieving their first independent funding. And several UCI Mind faculty members were involved in the new NIH and Alzheimer's Association-funded Impact AD program, a new national training program in clinical trials. Combined with our continued efforts in community outreach and community education, especially education that reaches Orange County's diverse population, I hope you will agree that our faculty, staff, and trainees are having tremendous positive influence on the next generation of dementia clinicians and scientists. But while education is an important part of our mission, research drives our mission. Our faculty continue to be among the most productive and successful on campus. Numerous faculty published highly visible and important discoveries in the last year in journals like Nature, the New England Journal of Medicine, and the JAMA journals. Funding from NIH and other sources has never been higher in the illustrious history of the Institute, and we are committed to discoveries that find solutions for Alzheimer's disease and related dementias. We do this as an important member of the national and international ADRD research community. And today, you'll hear from both UCI investigators and colleagues from around the country. I will be your guide on this 4-hour journey that will include presentations of cutting-edge findings from the impact of the coronavirus on Alzheimer's disease to the impact of the neighborhoods in which we live. Throughout the day, you'll also hear from the various sponsors and supporters who have helped make this conference possible. These sponsors include 24-hour home care, the Accord Study, Acticare, Active Care Residential Memory Care, Belmont Village, Cal Optima, Caring Companions at Home, Geiser-Patterson Law, Granny's Place, Independence at Home, Medi-Cal Consulting, Sorrentis Clinical Research, United Home Health, and Vivanti. We've tried to incorporate adequate breaks in the agenda, and we hope that you'll give the messages from these sponsors their due attention, as we are most grateful for their support and collaboration. And to get those messages started, here is a brief 1-minute spot from us at UCI Mind. And now let us turn to the first presentation of the day. No area of research came further, faster in the last year than did blood biomarkers for this disease. To describe these advances, we are quite pleased to be joined by Dr. Sid O'Brien from the University of North Texas Health Sciences Center in Fort Worth. Sid is a professor of pharmacology and neuroscience and the executive director of the Institute for Translational Research there. Sid is leading numerous extremely large biomarker efforts, in particular to understand AD biomarkers in populations that are historically underrepresented in Alzheimer's disease research, including Blacks, Hispanics, and people with Down syndrome. He has some 200 papers, and those papers have been cited more than 7,000 times. Let's hear from Dr. Sid O'Brien in his talk, Blood Tests for Alzheimer's Disease. Close, but still not there.
Welcome to my talk on blood tests for Alzheimer's disease, titled Close But Still Not There. So thank you for your organizers for having me here today. I think this is an incredibly important topic, obviously. And I hope after today we can realize where we're at, where we need to be. Some incredible strides have been taken, and what are the next steps that we need to get to. So disclosures, funding, etc. And I always like to start with the most important thing, and that is thanking the people who actually do the work. So this is at least a large list of most of the people in the Institute that I think deserve the credit for where we are today, as well as collaborators, which are far too many to actually list, so a few are there. Okay, so why even have this talk? Many of you, especially just in the last 2-3 years, will have seen just an explosion in research around biomarkers, blood-based biomarkers, in Alzheimer's disease, as well as other neurodegenerative diseases. And there's been some very exciting findings. I'm going to survey some of that today. A lot of this began, or at least began, a lot of the recent attention, a lot of that spurred on from the Nakamura 2018 article, which used a different way of assaying plasma amyloid species and came up with a composite biomarker that was very strongly related to Alzheimer's disease. And it's interesting because you can go back approximately 10 years to the Tony Muscaret 2007 Nature article that caused a very similar spur in research around this topic. So here we are in 18, and you'll see that there's just been a ton of things, of articles that have come subsequent to this. And here's just a few recent ones. These are all 2020 or 2021. And what's happened, as you'll notice from these, just these titles, is a broad range of things. A lot of interest in the newer p-tau species, still very strong interest in the plasma amyloid, and then also more proteomic profile approaches. But also what has been very exciting, if you look at the bottom two, is people looking at these across diverse populations, which you can see from our cohort, but as well from the YCAP Columbia research team in the cohort. So that's very exciting to see. And just some brief results, as the Ashton paper looking at p-tau 231 in a variety of different cohorts with the AUCs looking fairly promising. Our work in a multi-ethnic cohort, profiles of AD and MCI, very promising. Then the plasma amyloid work from Schindler around the C2N profile, also looking promising. So you see some really interesting nice findings. Here's another article where looking at the Biofinder 1 and 2 cohorts with a variety of things. And you'll see in here is that there's either p-tau217 alone or in combination with other markers. Typically in this work, combinations of markers outperform individual markers. Occasionally they're comparable, but normally the combinations perform better. And here's just a summary that I pulled together based on some of these recent articles. And you'll see the AUCs being very impressive. And this is just for AUCs around plasma markers detecting cerebral amyloid via PET or CSF. We'll get to the rest of that table in just a little bit. So what's changed? Why have we seen this just rapid expansion in the field? Well, I think primarily it's technological advances, so that there have been some pretty substantial strides taken in the technologies such that the assay platforms themselves, as one major technological advancement, have improved substantially. So able to detect much lower levels of abundance in proteins, but also more reliably. And as an example, my institute's biomarker core has run over 20,000 Simoa assays in the last few years, and the CVs are staying at around 5%. So the instruments are performing fairly well. The other is around the automation advancements, or not only advancements, but more consistent integration. And so what you're seeing is that, for example, there's the Simoa HDX, which is a standalone, fully automated instrument. Our institute has integrated 3 separate platforms into an automation system using Hamilton Robotics. And this is happening more and more, same with the instruments using the CSF. And what you see is with the automation procedures, performance also improves. But additionally, the throughput is getting better. And just as an example, we can run in our core 50,000 samples a year on each of the assays. So 1 sample on all 3. So we're talking hundreds of thousands of assays a year, which is a major change from just even 5 to 10 years ago. So these technological advances have not only improved the assays themselves, but improved the throughput with which we can do these things, so the data is turning around a lot faster as well. So why blood-based biomarkers? And I've been doing this long enough that arguing for why I think is pretty simple, but it's still not straightforward. But blood-based biomarkers are obviously less invasive and at least should be more cost-effective. They are more scalable. Now that depends on the platform that you're working on, but you can definitely do more blood draws than you can lumbar punctures in a year. Or PET scans. So definitely there's more scalability. There's also, though, especially for individuals in the lines of research that myself and others of you do, increase access to clinical research, clinical trials. There's still a major lack of representation of diverse communities in Alzheimer's research. And so implementation of things that can go into community-based studies have a lot of potential for increasing representation. One of the things that I've been arguing for almost over a decade, way longer than that, is I believe that blood-based biomarkers can increase access to confirmatory diagnostics, so those PET scans and lumbar punctures. Now, what's come around lately is, will blood-based biomarkers replace CSF and PET methods, which is something that is an interesting question. So we're going to dig into that one. One thing that I find interesting and sometimes a little fun is looking back, right? And this is a picture of a slide that I gave a talk in 2017 at AAIC, and talking about why blood-based biomarkers should at least be examined, because at that time there was a different pendulum was in a different place. So when I started, blood biomarkers were thought to have little to no use in Alzheimer's disease. Today now, what it looks like is blood biomarkers potentially have but the potential for replacing PET and CSF methods. Where I'm hoping we will be in the near future is somewhere in the middle, where these blood biomarkers are used as part of our armamentarium in a comprehensive set of tools. I don't think it's one or the other. So how do we do this? How do we take and advance discovery-based things towards clinic? And the biofluid-based biomarker professional interest area of the Alzheimer's Association in 2017 had a really amazing group come together that I was fortunate enough to lead this group to help try and lay these things out. And at that point, and still unfortunately in my opinion today, we're still kind of in the wild, wild west, where we see some just absolutely elegant design studies, some beautiful findings. They're still largely not being cross-validated in independent labs. The sample sizes oftentimes remain very small. Unfortunately, what's happening also, though, is some of these, knowing a lot of the people in this space, some of these things are not cross-validated, and then they're not published, and that doesn't help. One of the things that I think is another barrier to us advancing is a lack of presentation of the adequate data. What I see now when I survey the literature are AUCs without anything else. So confidence intervals around an AUC, but complete lack of sensitivity and specificity estimates. And, I think journal editors forgot about the STAR-D guidelines. And you can have an AUC that's 0.9, but sensitivity and specificity could be drastically different, even though that AUC is the same. An AUC does not tell you how to apply a test, nor do correlations. I think people forget that an AUC of 0.7 is still only approximately 50% shared variance. So when we're thinking of does something really relate strongly and surrogate for something, correlations under 0.95 are just not real, not useful. And so we need to see the actual data more. And so that's one thing that is something that we need to deal with. There are also significant methodological considerations that go beyond discovery. And I think one thing that we as scientists are great at is discovering things. So we create these wonderful assays and find these novel and very important findings. And then it stops, and it's just assumed that this is what it is and it can go to clinic, but that's not really how things work. Instead, we need to really be identifying the context of use ahead of time, rather than having studies that are biomarkers in search of a context of use, is how I view it. Once you define the context of use, it's a lot easier to design the studies and the outcomes correct, et cetera. And one of the things that we published in that 17 article was that basically what you see is the design of the studies in academic versus industry are completely backwards. So in an academic setting, and if you go look at almost every article that I'm going to show you, it's a case control study, correlations or some sort of mean group differences, and you find that there's this biomarker that's somehow different between cases and controls. And now we say this is a biomarker of disease. In industry, you define what you want the biomarker to be for, identify the fit for purpose, then you build the methods around that and you examine it. And this is why approximately 90% of academic findings don't hold in industry, because we don't follow through. And I don't think it's either or. What we believe is there's a comprehensive model that can get at this. But those articles that I showed you, pretty much all were along this line of case control. We find a correlation. Here's what we think our biomarker of. However, the different model is define that context of use. So it could be the discovery component is the first phase. But then we really must work on, let's define that context of use. Now we need to go into new studies. We need to confirm these things. We need to prospectively test them. We need to blindly test them. What's the cut scores? So there's a variety of ways and things that need to happen before something can make that all the way to the final clinical implementation. And it doesn't stop there. There still needs to examine performance even after it's in clinic. So from just that early question of biomarker for amyloid, cerebral amyloid. So can plasma biomarkers be used as a surrogate for PET and or CSF amyloid confirmatory diagnosis? So can plasma replace those? So here's those articles that I showed you. Now keep in mind that almost zero of these articles actually presented sensitivity and specificity estimates, so I had to go guesstimate them from the AUCs. They'll be pretty close, and I was generous. Now, clinical implementation requires some sort of knowledge around prevalence rates of whatever it is you're doing. So the first context of use that I'm putting the calculations in is for using blood to screen into a prevention trial. So estimated base rates of, say, amyloid cerebral positivity between 10, 20, and 30%. And this will depend on age. Well, as you'll see, if you take the first one with an AUC of 0.9 with a sensitivity and specificity of 0.9 and 0.75, if you go all the way to that 30% base rate, which is approximately the base rate in the A4 trial, you have a 61% positive predictive value. You have a 95% negative predictive value. And that's about as good as it gets. And so, at least in the context of use for detecting cerebral amyloid positivity for screening in something where base rate would be of anywhere as high as 30%, unfortunately, no. These markers are absolutely not surrogates, and they're not even close. What about for clinical diagnosis? Okay, so I took those that were looking at clinical from a primary care, from community-based, et cetera. And same base rates. Base rate of Alzheimer's clinically would be around 12% of those 65 and above. So if you're thinking of a primary care setting, go to that first column around a base rate of 10%. As you'll see, really none of them would perform well enough to be a true confirmatory diagnosis. And actually, most of them are not even very close. If you increase the base rates, they get better, but they're ruling out disease, not ruling in. But if you flip the context of use and we say, okay, what about for an Alzheimer's trial or Alzheimer's patients? And I want a neurology clinic where 50% of the patients are Alzheimer's disease. It's a non-Hispanic white population where we already know 70% approximately of those clinically diagnosed with amyloid in the brain. So how would it function then? All right, I took those same estimates, put them in the base rates for 60, 70, and 80%. And as you can see, your positive predictive value really drastically increases as a function of that base rate. However, your negative predictive power is now absent, largely absent. So basically, no, blood-based biomarkers are not surrogates for PET and or CSF diagnostics, and there's really no data to support that as a context of use. However, this is the same scenario with most complex diseases. There's a very nice article by Morgan et al. In 21 from JAMA Internal Medicine that looks at accuracy of practitioner estimates of disease probability based on testing of 4 common diseases: pneumonia, cardiac ischemia, breast cancer, and urinary tract infections. And what those authors found was that practitioners overestimate the probability of disease before and after testing, and widespread overestimates of the probability of disease are likely contribute to overdiagnosis and overuse. So basically, many clinicians, what we're doing is we're assuming the base rates in our clinics. So if we're in a memory disorders clinic, 50% of the people walking through the door have Alzheimer's disease, maybe higher. But that's a very select population. If you move this out into a primary care setting and it's only 12% of those 65 and above, you're going to overdiagnose disease most of the time. So how do primary care tests work? Well, primary care screening tests work as rule outs. So if you look at this figure, numerous primary care sorts of screening tests, the negative predictive values are very high. The positives are poor across the board. But that's because they're not used to diagnose. They're used to determine if something else needs to be done. So in a primary care setting, you use this blood glucose. If it screens positive, it means, well, you really should do something else. Does it mean that this person has diabetes? Well, it means they have a 50% chance of having diabetes. PSA is even lower. However, if you screen negative, likelihood is you do not have the disease or condition of interest. But what I'm seeing in some of these articles is people are saying things like, we're finding higher false positives than false negatives, and then making pathological interpretations. I think we need to be careful with these sorts of things, because we need to remember that amyloidosis happens in every organ in the body. And so it could simply be a function of the diagnostic accuracy statistics and how they're being applied. Screening tests are not supposed to be confirmatory. They're supposed to rule out as a first step. And so how do we frame these questions in a manner that might better leverage the power of blood-based biomarkers? And so these are different biomarker categories from FDA context of use examples: diagnostic, predictive, and response. And so I'm going to start with a context of use that I think that our group has been looking at for a long time, but there's numerous ways to look at it that I think could be very advantageous. So what about blood-based biomarkers is the first screening step in a multi-tiered diagnostic process. Well, that process looks just like cancer, diabetes, HIV. Pretty much every complex disease on the planet has a multi-tiered system just like this. And so we're just trying to mirror that. So blood-based as large-based screening tools. So, how do you get to where you can screen at that primary care so that primary care doc can refer over to the specialist? Once you get to the specialist has this huge armamentarium of things that he or she will do. However, how do we screen 40 million Americans from that first step? And keep in mind, most Alzheimer's patients are diagnosed and treated in primary care. They never see a specialist. And this is not unique, again, to Alzheimer's disease. So how do we meet that need? And that's something our group has spent a lot of time looking at. And that's just one context of use. That's not the end all be all. So we have the Alzheimer's disease in primary care study, which was designed as an example of that context of use. So we're enrolling 500 patients from primary care. And then they undergo the blood test, MRI, the PET scan, and then we'll test it. Does our blood test profile we created and validated work as a screener for primary care? But this whole model can also be done for screening into trials. So can you use blood-based markers within primary care settings to funnel patients into those clinical trials in the specialty clinics, which would be of tremendous value in increasing access to clinical trials, especially to diverse populations. As with all of these screening tests, the first step is rule out, not rule in. So rule out those 70% of individuals who don't need additional testing. And so as an example, if you can rule out 70% of people with a blood screen in primary care, then you could do phone interviews, MRIs, et cetera. You can arrange it however you want, but increasing access to get them to that specialty clinic. This increases that specialty clinic's pool of potential trial patients just exponentially. But it also does so at a lower cost. So this is a context of use that personally I think is incredibly important, and we're working on it as are other people. Just has to be tested in this manner. Now, another context of use that I know early on, many thought probably blood-based biomarkers would not work for, but it turns out it looks like that actually they may be very useful for. And I'm just throwing this one out there because there is some data on this of how do we target specific therapies to specific patients. And this is not unlike saying amyloid positive to an amyloid lowering drug. It's just more refined, looking for more refined subsets of patients. So for an example, neurodegenerative diseases, different neurodegenerative diseases, and then there's different subgroups of Alzheimer's patients. Well, we've already demonstrated that we can take these sorts of approaches and identify patients who respond to NSAIDs, to rosiglitazone, as well as duloxetine. For example, that the article we published in 2018 on NSAID therapy, which was, we demonstrated that a subgroup of patients that had elevated pro-inflammatory phenotype, and that's the group more likely to respond to an anti-inflammatory drug. And those are the markers that were involved. However, we also just found the same thing with REFLECT trials using rosiglitazone, and that could use a metabolic / inflammatory profile, which we have predicted because rosiglitazone has some anti-inflammatory properties also, and combine over those. Very accurate determining who responded and who didn't respond. And response for us is stable or improved over trial duration, not slower decline. So these are all patients who are either stable or improved. We've also now demonstrated this approach works in Parkinson's disease in a DataTop trial, the ADAP prevention trial, and we're currently running samples for pioglitazone in Parkinson's disease. So numerous trials now supporting the utility of the approach of blood-based biomarkers. Potentially, whether our approach is the approach that's going to work or not remains to be seen. I'm sure there's other ways of identifying these subgroups, but wanted to throw that out there that this is really under-examined in the space, and there's freezers full of samples to demonstrate the proof of concept to justify the prospective trials. The last context of use that I wanted to just throw out very briefly is surrogate outcomes. And this, for many of you, you realize is incredibly important now because the FDA's recent approval based on a surrogate outcome. The surrogate outcome used, amyloid, just isn't demonstrated to be a surrogate outcome. But I don't view that as a problem for anyone other than us. This is our job as a field. What we need to determine is, what are the surrogate outcomes? What surrogate outcome would change from baseline to 6 months that would predict that 24-month outcome? And I wholeheartedly believe that cerebral amyloid will do that in a subset of patients. But which patients? Is there a subset of patients that if you can quantify and identify them, that this type of patient, if you have this level of amyloid within 24 months, you're going to decline in this amount of time frame? Well, now you have some sort of argument of starting to really try and figure out, can you create a surrogate outcome at, say, 12 months, 6 months that predicts that 24, 48-month outcomes? And so many of us believe NFL might have utility in that. We have now data from the ADAP trial and others showing that change in some of these profiles, proteomic profiles, even at, for example, a 12-month change in ADAP predicting 48-month outcomes in ADAP. So still very early and very preliminary, but I think this is another area that really is understudied from a blood-based biomarker standpoint that could be of tremendous value to patients, to companies, to everybody. And this is something that I think we need to spend more attention on. So I know I've covered a lot of information. But in general, you know, I'm a very much glass half full kind of person. And I think blood-based biomarkers have a tremendous potential to advance the field towards more successful prevention and treatment options. We can help patients. However, I think blood-based biomarkers need to be put in the context of the available tools, and we need to stop trying to find one biomarker to do everything. I mean, a biomarker that detects disease may or may not be useful in predicting future risk. It may or may not be useful in predicting as a surrogate outcome. I mean, you know, for some reason in the Alzheimer's space, we have this tendency to want one thing to do everything. And you can look up plasma amyloid, you can look p-tau 181, p-tau 237. All of these markers, 217, we're seeing all of these contexts of uses being studied. And what we need to do is focus in. Where is it that we think this one helps, and how and why design the studies? So I think the field needs to think beyond just diagnostics as well. I think some of these markers have tremendous potential in other areas, and they just haven't been studied yet. But there's freezers full of pre-randomization samples from trials. Could look at patient selection sorts of things. You can look at surrogate outcomes. I think these markers really need to be looked at. And we need to spend more time refining the questions, focusing the questions. The discovery work is amazing. There's so many just incredibly impressive articles that have come out in just the last 12 months, much less 24 to 36 months. The vast majority, however, are discovery. They're very first pass early discovery work. So there's a lot of work that needs to happen beyond that. We need to take them and move them forward, because I believe these markers have tremendous potential to help patients. So I appreciate your time.
Okay. Dr. Sid O'Brien, thank you for that great talk, and thank you for joining us live now as part of this conference. Just so our audience knows, I'm getting questions relayed to me from Facebook, YouTube, and other channels. So if you have a question, whatever platform you're on, just type it into the chat, and it'll make it to me, and I'll ask Dr. O'Brien. Sid, thanks again. Really honored to have you join us as part of this conference. Maybe just to get us started, what would you say are the 2 or 3 most important pearls you hope our audience will take from your talk?
Yeah. So hello to everybody. Thanks for having me. Yeah, the talk probably I know it gets dense, and I apologize for that. I get a little carried away in the data sometimes. I think really the take-home point, in my opinion, is I think blood tests have a tremendous amount to offer patients, caregivers, docs, and over the last 5 to 10 years, there's been tremendous advancements. And in the last 24 months, even more. And I think we're definitely getting closer to having some forms of blood tests that will somehow help docs and patients find out what's going on. But I also, with that cautionary note of, I think we're just not there yet. And in some of the data I presented is very clear that even though some things look promising, we're just not there yet. But I think we're very close.
Yeah, I totally agree. It's been really exciting to see things come so far so fast. But
right.
Before you go into clinical practice, there's a lot that has to get done.
Right.
A couple of questions are coming in on the chat or through the various platforms. One of them is about whether blood tests can tell us about risk, in particular among people who might have a family history, who are concerned about getting Alzheimer's disease themselves.
Yeah, and I think that's one of the points of my talk is, what exactly do we need the test for? And that's where I think we need to spend more time as a field. So a blood test that would tell you that you're at risk, like a Framingham risk score for heart, right? That tells you, hey, you're at risk. We need to do something now to maybe do something about it. And there are people working on those sorts of things, including at UCI. And I think there's things like some people, were ApoE4 genes or whatever that do convey increased risk. At the moment, we don't have anything that I would say is clinically quite there yet, but that is one of the areas where I think blood tests are rapidly evolving, where I think within the next 5 years or so, we may actually have some tests that you can start taking in your 40s and 50s that tell you your risk, 10 years down the road, so that it can be actionable. And that's another point. We have to make sure that before a test like that hits the market, that the doctors that are using it don't just say, oh, you're at risk, now good luck. We have to have something actionable that they can do with it.
Yeah, absolutely. Stepping away from clinical practice for a minute and back into the research setting, right now, what do you think is the biggest value of the progress we've made in blood tests to our research endeavors?
I think where the blood tests are very soon going to be incredibly impactful is in clinical trial design. From a couple of perspectives. One, I think we will be able to use blood tests as a first step in a screening process to say who might be eligible for a trial. In the screening process, it's more likely that the blood test is going to say who's not eligible. And so whoever screens forward then gets more advanced stuff. And for some people, they wonder, well, why does that matter? Well, that matters because of cost. The screenings right now for these trials are enormous, but also access. So we work a lot with diverse communities. And so a blood test that can be in primary care to screen in the clinical trials, those who are running the trials would love it because they have much more access. Patients have increased access to the trials, et cetera. And a blood test that costs $200, well, that could be done across primary care clinics across the country. And so I think that's one. And another in the trial space that I think is coming soon would be looking at a blood test as an outcome variable. We're not there yet. I'm not going to say we are, but I do think we're getting closer because, in Alzheimer's clinical trials, and many people who are listening to me may have been part of trials or have loved ones in trials. These trials take a very long time. And the reason they take a long time is it takes a long time to determine if something impacts memory and thinking. But if we had some sort of test, blood test or otherwise, that said, hey, 6-month change in this marker tells me 24, 48, 56, however many month outcomes, well, now we can run trials in half the time or less. And that is huge for patients, but even for companies, because if they can know early that the trial's not working, we'll stop and move on to another one. It frees up money for other trials. And if it works, it gets to patients faster. And so I think that's to the science space. I think trials are where it's going to have the biggest impact soon.
Yeah, great answers. One of our UCI Mind faculty members, well known to you, Dr. Liz Head, asks, really appreciate your point about a single outcome versus a panel. What do you think would be in a panel or profile to give us all the information we need from blood?
Wow, Liz, that's tough. Hi, Liz. Good to hear from you. Yeah, and Liz, I've known Liz a long time. I'm a firm believer in profiles over individual tests because if you look outside of the Alzheimer's space, that's almost always what works better, even in the cancer space. I don't know that we'll ever have a blood profile that gives us everything we want, but I do think that we could very soon have panels of markers where you could run one test and get multiple blood tests out of it, right? So you do one blood draw and you get these 30 markers, and 5 of the markers are to say whether you're eligible for a trial. 5 other markers might tell you what trial you're eligible for. Should you go into an amyloid trial? Should you go to a neurodegeneration trial? So I think some of the newer markers like the p-taus and NFL are going to have a lot of potential in there. And like Dr. Gerl and I were talking about is even discriminating, right? This might be Alzheimer's or it might be a mixed pathology. And so I think that's one of the beauties of the blood profiles is you can capture so much that one blood draw, you might actually be able to run a whole suite of panels on one person. And now you're not just screening them far into a trial, but what arm of the trial. And then you've already got baseline measurements to know what changes over duration. So I think the newer markers are coming out are really impressive and useful, but I still think that, things like the inflammatory markers and metabolic markers, they're going to be key for certain subsets of patients. In those targeting who should get what drug. So I think it's exciting. I mean, it really is. It's exciting, and I think we're right there. And I do think the approach for the trials is going to be, we're not going to run one blood test. We're going to draw a blood tube, and we're going to run a whole bunch of things on everybody, because even if you're not eligible for this trial, that doesn't mean you're not eligible for this trial over here. And that's very valuable, and that's helpful for patients. I mean, if telling me I'm not eligible for something doesn't help, I mean, tell me what am I eligible for?
We've got about 2 minutes left. I'm going to throw 3 questions clustered at you.
Okay.
They're all in the clinical area. When are we going to get a test?
Okay.
Is it going to be covered by insurance? Mm-hmm. And what about other markers like saliva or other non-invasive types of tests for Alzheimer's?
So I think there is a test out there right now. It's not a primary care test, and I don't think it's useful for that. But I think a primary care test, a screening test, will probably be out within 24 to 36 months. I truly believe a screening test in primary care will be out there. I do think things like saliva and others are going to be useful. I think saliva may actually be incredibly useful for that long-term prediction, right? You catch them now, and where are we going with that? Okay, now I'm blanked on your third one. I got the first two.
It was about whether insurance would cover these.
Insurance coverage, more than likely, when something hits the patient market at first, insurance will not cover it. That's kind of normal stance. But after you demonstrate utility, I think insurance will, because of the cost containment that a blood test will provide. I mean, it will absolutely save patients money. It will save the insurance companies in the medical field money. So I do think it'll get covered, but I don't think it'll be covered straight out of the gate.
Great. We've got about 30 seconds left. One of the earlier questions from an investigator at UCI Mine was, these early markers, if you catch it early, does it lead to better survival or outcomes for patients?
That right there that's the goal. Can we find these markers that are really early, and can those markers also tell us what can we do from a prevention standpoint or an early intervention standpoint? And I think that's going to be where the biggest impact on patients will be. I'm one of those also. We have to treat our patients. You have Alzheimer's disease. We have to find better therapies. We also need to move earlier, because the best way to deal with the disease is going to be prevented. So I think we're there, and I think that's actually the biggest question right there, is how do we predict it? And like I said before, it has to be actionable.
Dr. O'Brien, fantastic talk. Thank you for joining us. Look forward to having you in 3 dimensions here in Orange County in the very near future. For our audience, we hope you enjoyed this talk. Stay tuned for a message from one of our sponsors.
Thank you.
My name is Gloria Barr. I have always been independent and thought myself quite capable. But now that I'm 92, I find that it's not as easy. I heard a lecture here at the clubhouse, and I signed up with Independence at Home. They have been terrific.
When I came to see Gloria, she was having some issues to open her pill bottles. I was just happy I was able to provide that to her and avoid any injuries.
I can open bottles now like Atlas. It's just a wonderful outreach to the community that I certainly would recommend to anyone.
Vivante is a special new type of senior living community. Dedicated to the highest quality. Created, owned, and managed by members of our local community. Where we celebrate our heroes. At Vivante, we're committed to the health, well-being, and happiness of all of our residents. Come be part of the best.
Okay, welcome back. It's time for our next talk, which will come from Dr. Dina Dubal. Dr. Dubal is on the faculty in the Department of Neurology at the University of California, San Francisco. She is the endowed chair in aging and neurodegenerative disease. Before that, she was a recipient of the illustrious Paul Beeson Scholar Award for early career faculty. Since then, she's done incredible work that's been featured in NPR, Time Magazine, The Economist, and The New York Times. She runs a lab. She's also a clinician scientist at UCSF. She's among the editorial leadership at the journal JAMA Neurology. And her work has featured two main thrusts, one of which she's going to talk about today, exploring sex differences in Alzheimer's disease and the possibility of protective genes. Enjoy this talk.
Hi, I'm Dr. Dina Dubal at the University of California, San Francisco. I'm delighted to join you today, the annual meeting at UCI Mind on Alzheimer's disease. We'll be exploring sex differences in Alzheimer's disease and the possibility of protective genes. I'm a neurologist and I'm a neuroscientist, and I direct a laboratory understanding the resilience of the brain in aging and Alzheimer's disease. And the people that you see in this picture sharing food pre-COVID is a group that leads really the discovery at the front line. So again, delighted to join you. We'll just jump right in. First, I have disclosures and relevant relationships. Disclosures are that I have consulted for Unity Biotechnology, an anti-aging company. I am an associate editor at JAMA Neurology, an academic journal, and I'm funded by the National Institutes of Health, including of Neurologic Disease of Aging, by the American Federation for Aging Research, the Simons Foundation, and I also receive philanthropy from the following families. We study resilience in brain aging and neurodegenerative disease. And resilience means that even though the toxicities are present, the stress is present, aging assaults the brain, Alzheimer's assaults the brain, that in some people, the effects of disease are minimal and there is resilience. And so if we can understand resilience and what underlies the biology of resilience, maybe there could be new therapies to counter this and defeat devastating diseases like Alzheimer's. We study Klotho, a longevity factor, which we will not be discussing today. We also study sex chromosomes in aging and Alzheimer's disease, which is the focus of our exploration today. First, I'll ask a basic question, and that is, why does sex biology matter? We'll be talking about sex differences. Well, it's because every cell has a sex that is either XX or XY in most cases. Sex biology influences fundamental processes through hormones, through the dose of the X chromosome. Females have 2 X chromosomes, males have 1, the Y dose, males have a Y chromosomes, females don't, through a process called female mosaicism, in which in every female cell, either the X from the mom or the X inherited from the dad is expressed because of random X inactivation that we'll discuss later. Now, there are true sex-based differences in aging and neurologic disease. For example, multiple sclerosis and Alzheimer's disease is more prevalent or more common in women. But did you know that women actually live longer with Alzheimer's disease, ALS, Parkinson's disease, and Lewy body dementia? Now, more than being just interesting that there are male-female differences in these degenerative diseases and in aging, understanding the differences could mean novel therapies. If we can understand the mechanisms or the underlying biology that protects one sex, that could be harnessed to treat both sexes. This is why sex biology matters. I want to show you a dramatic difference, a dramatic sex difference that is seen across the world in every society that records mortality. This is data collected by the World Health Organization, and life expectancy of women is graphed here on the y-axis, and life expectancy of men is here on the x-axis. And what you can see is that in every society, from Sierra Leone to Japan, every society that records mortality, women live longer than men. And so their lifespans are longer and above this blue line, which would indicate equivalent lifespans between males and females. So this is remarkable. And this is true and conserved during times of famine, of epidemics, and again, observed across the world. Truly a remarkable sex difference between males and females. Women live longer than men. Now, females will show increased lifespan across the animal kingdom, from lions to whales, not in every species, but in many, many species. Female lions live longer than male lions, and female seals live longer than male seals, and female whales live longer than male whales, as you can see here in this graph of survival, showing that female whales, here in red, show more survival than male whales, shown here in blue. What this graph further shows is that if the moms live longer, then so do the kids. And this is an evolutionary reason why it's helpful for women to live longer, that their offspring have better survival, but also their grandchildren will have better survival. And so there is an evolutionary advantage to females living longer. Now, what about mice? We study models of aging and Alzheimer's disease in the laboratory to the goal of new therapies for this devastating condition. But let's first ask, do female mice live longer than male mice? This is work done by graduate student Emily Davis. And what she found is that female mice, here in red and shown in this lifespan curve that are XX, live longer, have more survival compared to the male mice that are XY, shown here in blue, that progress to death faster in normal aging. And so we see a conservation. We see it in mice, something very similar to what is observed robustly in human populations. Now, when we try to understand sex differences, what makes a difference between males and females, one of the main differences is that females have different sex chromosomes than males, and they also have different gonads than males. So we first can ask the question, is it the sex chromosomes or is it the gonads, the ovaries versus testes, that can cause female longevity? In order to attack this question, Emily used a really interesting model. that I'll step you through, and put your Sudoku hats on. This is a really neat approach that is mental gymnastics. So female mice are XX, and then they also have autosomes, non-sex chromosomes. And male mice are XY, and they have autosomes, which are non-sex chromosomes. Male mice have a gene on their Y chromosome called SRY, and that differentiates a male into a male with testes, testosterone, and a male phenotype. Now, in this model, SRY is actually on the autosome. And so through the genetic gymnastics, we can create 4 sex genotypes: XX mice with ovaries, XY mice that are missing that SRY, that by default will develop ovaries, XX mice with the SRY that's on the autosome now that develop testes. These are male mice, they only have an XX genotype, and XY mice with testes, the typical combination. So if mice with ovaries live longer, those are circled, than the ones with testes, then that would mean gonads-mediated female longevity. But there's another outcome. If XX mice, circled, regardless of growing up with ovaries or testes, lived longer than XY mice, then this would be a sex chromosome-mediated effect on female longevity. So which one is it? Is it the gonads or is it the sex chromosomes? Emily found that it was sex chromosomes that contributed the main statistical effect to female longevity. So what she found is that XX mice that grew up with ovaries or testes didn't matter, shown here in black. They lived longer in this lifespan curve, you can see it, compared to the XY mice with ovaries or testes didn't matter, here in gray. So XX mice with XX sex chromosomes live longer. Now, does female longevity extend to degenerative diseases like Alzheimer's disease? Now, let's take a moment to understand the sex differences of Alzheimer's disease. How is it different in men and women? More women have Alzheimer's disease. 2/3 of people living with Alzheimer's disease are women. Now, we thought it was because women are more vulnerable to the disease, but it turns out that men and women have a similar risk of getting Alzheimer's disease until a very old age, until after the age of 80. Of note, men generally live to the mid-70s in the US. But we now know that women live longer with Alzheimer's disease. And when women live longer with Alzheimer's disease, there's more women with the disease. So here is an illustration. You can see that these are women on the left, XX, and men here on the right, XY. And the ones with skeletal faces are dying from Alzheimer's disease because men will die faster from Alzheimer's disease. And so what you end up with is more women here, XX, that are living with Alzheimer's disease compared to men. But there is resilience there because women can have the disease but live longer with it. Now, here it is shown in the graph form that in the lifespan curve that women with Alzheimer's disease here in red will live longer than men with Alzheimer's disease here in blue. Now, I want to make the note that we do not aim to have disease for longer and be miserable. That is not the goal of the research. However, if we can understand what helps someone to resist some of the disease and not succumb to mortality, if we can understand that resilience, there may be new therapies for both men and women that are derived from female longevity. And that is the very new thought and a very new approach here. So let's ask the question, do animal models of Alzheimer's disease recapitulate that female resilience that we see in humans? And just like humans with Alzheimer's disease, female mice that model Alzheimer's disease will live longer. And you can see here in this lifespan curve that in red, the female mice that model Alzheimer's disease live longer than the male mice that model Alzheimer's disease here in blue. Absolutely remarkable and mirrors the human condition. Now, there is this female resilience in the Alzheimer's mice. I'm going to call them human APP mice. And this resilience you can see in their cognition. You can see it at all stages of their life and different types of Alzheimer's models. And despite similar levels of Alzheimer's toxins between males and females. So, what underlies that resilience to Alzheimer's disease in male and female female Alzheimer's mice? This is work done by Lauren Brostel and Patricia Warden. And what they did was they applied that model that I explained earlier to Alzheimer's mice. And they see that as expected, XX Alzheimer's mice live longer than XY in blue. But absolutely remarkably, if the gonads were switched, XX mice still live longer. That is here, XX mice here in yellow, but these mice grew up with testes. They still live longer than the XY mice if they grew up with ovaries. So what you can see here is that XX mice that grew up with ovaries or testes didn't matter. The XX mice that model Alzheimer's disease will live longer than the XY mice that model Alzheimer's disease. And the gonads didn't make that much of a difference. It was really the sex chromosomes. So the question becomes, how do two X chromosomes confer resilience? And this is work done by Dan Wong, Casey Shaw, Emily Davis, and Samira Abdullah Saiku. Now, first, a basic piece of biology, and that is when an XX individual is developing, early on in development, there are two X's that are active, but very early on, one X gets shut down through random X inactivation. And therefore, female cells each have one X, either the X from their mom or the X from their dad. It's totally random, but one X is shut down. Now, we used to think that this shut down or silenced X was completely silent, but it's not. There is some escape where some genes are expressed in cells with two X's in female cells. Now, we looked at what are those genes or factors or proteins that are that escape that silent X in both humans and in mice. And there are really only a handful. And we studied, which I'll be talking about further, KDM6A. So the first question we asked is, does KDM6A, this protein you'll learn about, escape that silent X in mouse neurons? And this was work done in collaboration with Barbara Panning, an investigator here at UCSF. And what we found is beautiful, absolutely remarkable. You can see in this XX nucleus where the DNA resides of a mouse neuron, what you can see is that KDM6A is being transcribed, is being produced off of the active X, but also the silent X here, two places, whereas in males, it's only being produced from one X. What is KDM6A, also known as UTX? It's something called a demethylase, and that is a type of protein that unwinds the DNA of our cells and then allows for new RNA and proteins to be made, for genes to be expressed. There are human mutations in KDM6A that include intellectual disability. And this gave us the hint that KDM6A may be important for cognition in Alzheimer's disease. So we first asked the question, looking at human populations, does KDM6A expression levels, are they higher in the female compared to the male brain? This is work done spearheaded by Dr. Jennifer Yokoyama and Luke Bonham. Dr. Yokoyama leads a neurogenetics unit here at UCSF. So what they found was, indeed, in women and females, whether or not they had Alzheimer's disease, so without or with Alzheimer's disease, females had more of this protein, KDM6A, in their brains compared to males. And that is because females have 2 X chromosomes, one is silenced, but KDM6A escapes that silencing. So there's more of it in a female brain. Now, do higher KDM6A levels in the brain influence human cognition? Key question. So this is really fascinating data. What we found was that everyone has the gene KDM6A, but some folks have a slightly different code of KDM6A. And there is a certain code for KDM6A that makes it higher in your brain. So those people with higher levels of KDM6A, because they carry the genetic code, did not show cognitive decline here in blue over a decade. This is using an Alzheimer's database. Now, in brown, those people without this genetic code showed significant cognitive decline over a decade here in brown. So having that genetic code that gives one higher KDM6A levels associated with better cognitive function, resilience against cognitive decline, telling us KDM6A is important in the human brain. Now, getting back to mice, we can really ask questions around causation. Can we increase KDM6A in the male brain to confer resilience, to make it similar to the female brain? So what Emily and Samira did was some genetic engineering. This is a mouse brain, and they injected something into the mouse brain that incorporates more KDM6A and has the brain make more KDM6A in the male brain. So here you see just the control mouse, and here in the male brain, this lentiviral vector, a form of genetic engineering, increased KDM6A in the male brain. And what they found was absolutely amazing, that in the Alzheimer's mice, in the XY Alzheimer's mice, the memory impairment shown here in the white bar, outlined in blue, is very high compared to control mice. It's very high. But if KDM6A was boosted in the male brain, the cognitive impairment was reversed down to normal levels. So boosting KDM6A in the male Alzheimer's model brain really reversed the cognitive deficits. Now, Casey Shaw, a graduate student in the lab, was really curious about how does KDM6A act in the brain? How is it inducing resilience? Does its location in the brain cell matter? So what Casey did was to create a whole bunch of different types of KDM6A protein, shown here. She overexpressed it. That's what OE means. She mutated a nuclear localization signal, which means that it gets trapped outside the nucleus. It can't get into the nucleus. And then she mutated the nuclear export signal, which means it gets trapped in the nucleus, which you can see here. So she made different types of KDM6A to control the location of this protein within the cell. And what she found is that, well, these are ongoing experiments, but one finding is that, remember how we said that it's a demethylase, it unwinds the DNA and allows genes to be expressed? Well, when she got rid of that methylase function, it still induced resilience, meaning that she's finding a novel function of KDM6A in the brain. And she's also finding that it hangs out in the cytoplasm, which is the fluid all around the nucleus of the cell. And she is discovering what it's doing there to induce resilience in the brain. So we'll stay tuned on how KDM6A is inducing resilience. So in summary, I've shown you that women live longer in normal aging and in Alzheimer's disease, that aging mice and mouse models of Alzheimer's disease recapitulate that sexual dimorphism, and that women are not simply small men. Women and men are different, and there are different vulnerabilities and resilience. Number two, that sex chromosomes contribute female resilience in aging mice and in APP mice. And through those models of sex biology, I showed you that this has to do with having a second X chromosome. That second X really matters. And finally, that the X chromosome may harbor novel therapeutic targets, such as KDM6A, for diseases of the aging brain in men and in women. And so we're very excited to be delving into what the second X chromosome does. It's not just silent. There are genes being transcribed like KDM6A, and we wonder whether these could be new therapeutic targets. Acknowledgements include my lab, with folks highlighted in red who spearheaded many of the studies, collaborators, including Barbara Panning, who I mentioned, and Jennifer Yokoyama, people throughout universities, and as I had mentioned earlier, funding through several agencies. Thank you so much for your attention, and I welcome your questions.
Okay, we are joined live now by Dr. Dina Dubal. Dr. Dubal, thank you so much for that fantastic talk and your elegant work, and for joining us today live to take questions.
Such a pleasure to be here. Thank you for the invitation.
I'm going to start with a question about some of the data you showed, specifically the longitudinal human data and the, if you will, the gene dosing studies. And I'm just curious if the effects that you guys observed look the same in men and women participants in that study.
Great question. Thank you for asking it. So you're referring to the clinical observational study that had almost 800 people. This is from the ADNI cohort. And what we found there, again, just as a quick recap, is that certain people have a genetic code that gives them higher levels of KDM6A in the brain, and those people did not show cognitive decline compared to people without that genetic code. And when we looked at this data with several angles. First, we asked men compared to women, was the effect same? And the answer is that in women, we saw a very clear effect of this genetic code in conferring resilience. In men, we saw the trend, but our statistics were limiting in that we didn't have the power to make a definitive conclusion. However, when we grouped men and women together, the effects were quite clear that this genetic code conferred resilience. Another interesting point is that this population of around 800 individuals is really enriched for those with mild cognitive impairment, those that are really teetering on the risk of developing Alzheimer's disease. And it is in those individuals where we really saw a marked association with protection in this genetic variant.
Interesting. So mechanistically then, do you think this might be related to the pathology we associate with Alzheimer's disease, or something else that's happening in the brain?
It's hard to know, and that's really where our research is taking us right now. In our animal models, that the amount of Alzheimer disease toxin is the same with and without KDM6A. You can dial KDM6A up and the toxin levels are the same, but the ability of the brain cells and of the mouse brains to withstand the Alzheimer's toxins increases. So the brains become resilient even though the same amount of toxicity is there.
Great. Well, we'll look forward to more of that mechanistic research as it comes from your lab. There was a question submitted by one of our viewers. How does hormone therapy used within transgender communities in particular affect longevity and predisposition for disease?
Mhm. Well, that's a fascinating question, and one whose time has come. And the bottom line is that we don't know yet. This sort of information is really just now become of very high value and is now beginning to be collected. And so we wondered the same, but there wasn't enough information out there yet to know. But we could speculate. We could speculate that if what we see in our models could translate to the human condition, it doesn't always do that. But if it did, we would speculate that there would be some protective effect of female hormones, even in men. Again, pure speculation, but maybe ovarian hormones could extend lifespan in men. And as far as the risk of Alzheimer's disease, again, we don't know. But if there is some translation from mouse to human, maybe there would be some protection.
Great. Another question from our audience. Could you talk a bit more about subcellular distribution of these markers like KDM? And I'll add, could you also talk about potential regional differences throughout the brain in expression of these markers?
Mm-hmm. We're in the process of mapping out in a very anatomic manner, where is Kdm6A expressed? Is it in only certain brain cells but not in others? Is it in certain regions of the brain and not in others? We're doing this both in mouse brains and in human Alzheimer's brains. We do know that there is a very high level of this Kdm6A resilience protein in the region of the brain called the hippocampus, in the main cells of the hippocampus and the main brain cells of the hippocampus that are responsible for learning and memory. So they're in a prime real estate location. And this is, of course, important because Alzheimer's disease and aging really erodes learning and memory. So if Kdm6A is in these cells that are responsible for learning and memory, maybe it could preserve them over time and with disease. Now, there was another, really a puzzle and an enigma that arose in our research, which I spoke of toward the end of my talk, which is that where Kdm6A is in the brain cell is really fascinating because we know Kdm6A to be this, what we call demethylase. It opens up the DNA and allows genes to be expressed. But if it was really only doing that, it should be in the nucleus of the cell, which is the heart of the cell where the DNA lives, where our genetic code lives. But interestingly, we didn't find Kdm6A really stored in that powerhouse nucleus of the cell. Instead, we saw it in the cytoplasm, in the part of the cell that surrounds the nucleus. And that has given us a clue that it might be doing something else besides unwinding the DNA. So that subcellular location, where it is within the cell, is going to give us more information about what it is actually doing to make a brain cell resilient. That's where our research is going.
Yeah, that's great. We got like an immediate follow-up on the chat about asking you to speak about other cell types in the brain like microglia and astrocytes and whether there might be a role for those cells in this story.
Most certainly there is, and we just don't know what those roles are yet because we haven't gotten there. I think this work is fresh off the press, but you know, all these, the microglia, the astrocytes, the oligodendrocytes, neurons, they work together and they don't exist in isolation. And so I really appreciate the question. And Kdm6A is expressed in those cell types too. And so how they work together to produce resilience is to come. We don't know, but certainly the other cell types will play a role.
We have just under 2 minutes. I'm going to combine two questions here. What's the impact of menopause in this story, as well as what lifestyle factors or choices that men or women make could help them, increase their resilience or even, back down to the subcellular level, enhance this pathway?
Yeah. Well, I mean, good news. We know this from so much research from around the world that what is good habits make for a healthy brain. And what is good for the heart is good for the brain. As just a rule of thumb, exercise, social networking, a great diet, keeping the mind stimulated, all of these factors that we know to be a healthy lifestyle are good for the brain and really help resilience. Do they increase KDM6A? Maybe, we don't know. I think that's a brilliant line of thinking. We don't know. Menopause. So menopause happens in the early 50s, and hormone levels dramatically decrease in women. The good news is that our research is finding that even though the hormones were taken away, the effects of KDM6A were still strong. They didn't necessarily depend upon the hormone, although hormones can help with longevity. So I would speculate that even throughout and beyond the menopause, KDM6A could be potentially this resilience factor for women, maybe even men.
Dr. Dubal, these question and answer periods go so quickly.
Yes.
This has been fascinating and excellent. Thank you again for joining us and bringing your great work to this year's conference.
Such a pleasure. Thank you so much for having me.
And for our audience, stay tuned for another message from our sponsors, and then we'll have another exciting talk.
You've spent a lifetime making memories. We invite you to make more. At Belmont Village, residents find the perfect balance of exceptional living and exceptional support. A lifestyle to suit you, and for couples who need different levels of care. An experience designed by experts in hospitality, dining, architecture, and enrichment. It's not just the life you need, it's the life you've earned. Belmont Village Senior Living, the community built for life. You've spent a lifetime making memories. We invite you to make more. At Belmont Village, residents find the perfect balance of exceptional living and exceptional support. A lifestyle to suit you, and for couples who need different levels of care. An experience designed by experts in hospitality, dining, architecture, and enrichment. It's not just the life you need, it's the life you've earned. Belmont Village Senior Living, the community built for life.
I'm Diane Mondini. I'm president of Caring Companions at Home. I got involved in Alzheimer's OC back in the late 90s when my good friend Julie David served on the board and encouraged me to help with the galas. Because of my business and dealing with seniors for the last 30 odd years, I have seen how this disease has taken its toll on many of my clients and many of my clients' family members. It's very hard seeing them suffer and wondering what you can possibly do to help them. That's why I have a passion for trying to find a cure for this disease, or helping in any way I can.
Okay, our next talk comes to us from Professor Tom Lane from right here at UC Irvine. I happen to know that Tom began his academic career at Ball State University in Indiana before coming out west to do his PhD in microbiology and immunology at UCLA. For the most part, he saw the light and stayed in California, did his postdoctoral training at the Scripps Institute, was a faculty member at UCI, brief stint as a faculty member at Utah before rejoining us here at UCI, where he is now a chancellor's professor in neurobiology and behavior. Tom is an expert in a virus impact on the brain, has had funding as it relates to MS as well as other neurodegenerative disease. He's a member of the American Association for the Advancement of Science since 2020. And he's going to deliver a talk that I'm sure many of you are highly anticipating: viral infections and Alzheimer's disease. Enjoy.
Okay, I'd first like to thank the organizers for the opportunity to talk about some of the work we're doing in the laboratory. And specifically, we're very interested in how viral infections influence disease progression in patients with Alzheimer's disease. And specifically, what are the implications for COVID-19 on Alzheimer's disease pathogenesis? And so right now, within the past year, a prominent goal in our laboratory specifically is to determine if coronavirus infection increases neurologic disease in Alzheimer's disease mouse models, as well as in COVID-19 infected AD patients. And so this talk will probably be a little different than some of the talks you've heard today. I'm going to begin by giving a background on coronaviruses, COVID-19, and neurologic disease associated with SARS-CoV-2 infection, and then address some of the ongoing work that we're doing right now in my laboratory, which is specifically examining how coronavirus infection affects neurologic disease in mouse models of Alzheimer's disease. And then I'd like to conclude by discussing some of the future work we're going to be doing in the laboratory, and which focuses on examining how COVID-19 affects neuropathology in patients with dementia or Alzheimer's disease. And so, as you probably all are aware, coronaviruses are viruses that we've been living with forever, basically, all right? And there are actually a number of different coronaviruses that have the ability to infect and replicate within the central nervous system. And that's shown here, all right? You have mouse or murine coronaviruses that can readily infect and replicate in the brains of mice that cause a variety of different diseases. And actually, for the past 25 years, my laboratory has been focusing on looking at murine coronavirus infection of the central nervous system to better understand how immune mechanisms contribute to both defense as well as disease. There are also coronaviruses that infect cats that can cause neurologic disease as well as pigs. But up until now, the most common coronaviruses that we've all been associated with are the common cold coronaviruses, which are shown down here. And so two of these were identified in the 1960s. And then after SARS-CoV-1, which occurred in 2002 to 2003, there were two additional common cold coronaviruses that were identified in Hong Kong as well as in the Netherlands. And what these are basically viruses that are seasonal in nature and cause mild upper respiratory tract infection. So about 15 to 20% of quote unquote common colds are associated with these coronaviruses. But as of January of 2020, we're all very familiar now with another coronavirus, which has been identified as SARS-CoV-2, which is the etiologic agent for COVID-19. And this was a wonderful article written by a good friend of mine, Stan Perlman at the University of Iowa, which really captures it. Another decade, another coronavirus. And what he's alluding to is simply the fact that we had SARS-CoV-1, which occurred in 2002 to 2003. And that was primarily in Canada and some parts of Europe. And then in 2012, we had MERS coronavirus, or Middle Eastern Respiratory Syndrome. And this actually is still ongoing. But it's largely confined to the Middle East or the Saudi Arabian Peninsula. And then as of January 2020, as we all know, we have the emergence of SARS-CoV-2, which causes COVID-19. And so this virus is a little different than SARS-CoV-1 and MERS-CoV in the fact that it's much more dangerous and it spreads very well. But it also has a variety of other different clinical symptoms that are not restricted only to the lungs, which is very common with SARS-CoV-1 and MERS-CoV. But with COVID-19, heart disease can be a very common problem. Kidney damage has been identified. You have inflammation of the blood vessels, which can lead to stroke in some patients. You have hyperinflammatory syndromes, which has been reported in children. And then, interestingly, you have asymptomatic spreaders. And this isn't as discussed as much now as it was when the virus first came out. But essentially, one of the big differences between SARS-CoV-2 and MERS-CoV and SARS-1 is simply you can be asymptomatic, but infected, and then spread the virus very efficiently. So you don't know you're infected, right? You don't have any of the classic symptoms that are associated with upper respiratory tract infections. And then finally, there is, without a doubt, an increased frequency of neurologic disease or conditions that are associated with SARS-CoV-2 infection, which was not as common with SARS-1 and MERS-CoV. And this allows a springboard into something that I believe is very important and needs to be examined in great detail, and that is COVID long haulers, all right? And I assume that most of the audience is familiar with this. But essentially, it's novel symptoms that are associated with infection with COVID-19 or SARS-CoV-2. People have cleared the virus, but symptoms remain for extended periods of time. And so again, these are referred to as long haulers. They often test negative for the virus. Doesn't mean that the virus may not be there, but they definitely test negative via PCR. And right now, the evidence argues that about 10 to 15% of COVID-19 patients become long haulers. That number varies based on the report. And keep in mind, the disease, COVID-19 is still a very young disease, and we're still learning quite a bit about it, all right? But long hauler symptoms can affect anyone, young or old, healthy, no comorbidities. And interestingly, it's been detected or identified in patients that either had very severe COVID-19 symptoms, so they were hospitalized and placed in an intensive care unit, or in those individuals that had very mild or benign symptoms. All right, so there's no correlation with the severity of disease and the ability to develop long hauler symptoms. Neurologic disease that's often commonly associated with long haulers include headaches. They can have a loss of taste, of smell, even if they didn't have it when they were initially exposed to the virus. Most common neurologic conditions are brain fog, all right? And this can be identified with cognitive decline, memory issues, difficulties in concentrating, and then this also is associated with chronic fatigue syndrome, all right? And there's an interesting article that just recently came out in Nature, which is a very prestigious journal, by Tony Weiss-Corey's group at Stanford that identified that in some COVID-19 patients in which they could identify virus in the brain, there were different areas of the brain that the virus could be detected, and they found that the presence of the virus disturbed a constellation of genes that were important in controlling cognition, as well as associated with schizophrenia and depression, which is interesting. I think this is an area of research that needs to be pursued in more detail. But getting back to some of the neurologic factors, all right? And so this cartoon kind of sums up what we all know, that, again, COVID-19, it's a respiratory disease. Virus is inhaled, and the virus prominently goes to the lungs and can cause a variety of symptoms in the lungs, more severe being pneumonia and ARDS, which is an acute respiratory distress syndrome. But again, you get neurologic symptoms that can be the most common being loss of sense of taste and smell, and headache. Those are most commonly reported. However, in more severe cases, you can get seizures or encephalitis, which is inflammation of the brain, which is immune cells going into the brain following infection, and then sometimes an altered mental status, right? The global incidence of neurologic symptoms in COVID-19 is about 80% of all hospitalized patients have reported some component of a neurologic effect associated with the virus. Again, going back to the notion of long hauler syndrome, 6 weeks post-infection, a variety or a number of patients have reported this brain fog. And then more recently, there was an intriguing article that shows that a loss of gray matter or brain tissue in patients following infection in non-hospitalized patients. So this is intriguing, but needs to be followed up on. So if you look in the brains of postmortem patients that succumb to COVID, surprisingly, there's not robust infection of the brain, at least at the time of these individuals passing, but you nonetheless can detect virus either by sophisticated molecular techniques called PCR, where you amplify viral RNA, or you can go into the tissue itself and look for viral proteins. So this is this staining, if you will, for the spike viral protein. So spike is that projection from the surface of the virion that allows attachment to host cells. And you can see clearly here these neurons that are infected with virus as detected by expression of spike. Some of the most prominent neuropathological findings that are observed in patients with that have succumbed to COVID include activation of resident cells of the central nervous system, including microglia. These are, again, the immune cells of the central nervous system, as well as astrocytes. So these cells get activated, and they have a very distinct morphology and expression of different genes. In addition, you can see immune cells or lymphocytes infiltrating into the central nervous system of these patients. And also, one of the more common features are these acute ischemic changes, so lack of oxygen, right? And that's probably associated in more severe patients that have severe respiratory distress. So the first question that we were interested in, and this dovetails beautifully in our work that we've been doing with mouse coronaviruses, was it's a very simple question. Is SARS-CoV-2 able to infect and replicate in human neurons? And so we did a very straightforward experiment. We cultured human neurons in the laboratory in these little culture dishes. And then we infected these cells with SARS-CoV-2. And then we waited a defined period of time, so 24 or 40 hours post-infection. And then we just simply looked to see if these human cells were susceptible to infection with virus. And indeed they are. And so these are some beautiful images that were acquired by a wonderful graduate student in my laboratory, Hema Oliveira. And what we're looking at here are neurons stained in red. And what that red is, it's simply we're looking at a protein that's expressed in neurons so we can identify these cells. All right. And MAP2 is this protein. Okay. So all neurons express this. And so 24 hours following infection, then we went in and looked for SARS. Okay, I'd first like to thank the organizers for the opportunity to talk about some of the work we're doing in the laboratory. And specifically, we're very interested in how viral infection There's plenty of virus in the cell or the soma of the neuron. It's not in the nucleus, which is coronaviruses don't go into the nucleus of cells. But by 48 hours, we can see the virus is starting to spread down these dendrites, or these long extensions that neurons classically have. And you can see it also here under low power magnification, but very nicely right here, this virus going down these neurons. And I think this is actually a very interesting question that we're pursuing. So you might have a low level infection in the brain following exposure to SARS-CoV-2. And is this how the virus spreads in the brain? It doesn't replicate very extensively, but does it use these neurites or extensions from the cell body to spread? So we're starting to look at that in more detail. In addition, what I'm not showing, but I just thought I'd mention this, we also looked at the molecular level that upon infection of these neurons, neurons, like all cells, if they're infected by viruses, it elicits a very rapid and robust expression of genes that encode for antiviral proteins, and this does occur in neurons. All right, so we know neurons can be infected by SARS-CoV-2. This complements a body of literature that has also indicated this. And the next thing we wanted to do in the laboratory was develop an infection model of mice with COVID-19 so we could better understand mechanisms contributing to the pathogenesis of disease, as well as neurologic consequences of infection. Okay. So what we did is what a number of other laboratories around the world have done. We tried to develop this, a preclinical animal model. All right. So one of the problems very quickly is you all by now, I'm sure all of us could have a very nuanced conversation about SARS-CoV-2 pathogenesis, but the virus uses a receptor to gain access into cells in humans, and that receptor is a protein called angiotensin converting enzyme 2, or ACE2. Okay, so one of the problems is the mouse is the most common animal model for almost all viral pathogenesis studies, and mice express ACE2, but their ACE2 cannot bind SARS-CoV-2. All right, or in other words, the spike glycoprotein of SARS-CoV-2 cannot recognize and bind to mouse ACE2. So if you want to work with mice, you have to engineer the animals to express human ACE2. And there's several ways you can do this. One, you can actually use other viruses to deliver. You can construct these viruses to express human ACE2, and then you can use these viruses to infect mice. And then unbelievably, those animals then express human ACE2, and they're susceptible to infection. Alternatively, you can engineer mice to simply express human ACE2 in the lungs and other tissues in the body. And so these are called transgenic mice that express human, or ACE2, okay? Alternatively, other laboratories have used non-human primates, so monkeys. The monkeys do express a human ACE2, for lack of a better description, so they are naturally susceptible to infection. And then you also have Syrian hamsters, which remarkably express essentially an ACE2 that's identical to humans, so you can naturally infect those animals. But our work, we've been doing mouse pathogenesis work for over 20 years with mouse coronaviruses, so we wanted to go with the mice. So we've been employing transgenic human ACE2 expressing mice. And what we can do is we can intranasally infect these animals, and they get a beautiful lung infection consistent with what you see in humans. But again, we're more interested in what happens in the brain. So we can remove the brains of these from animals at defined times post-infection. And specifically what we're interested in is viral infection of the brain or the spinal cord, neuropathology, or in other words, disease or changes that occur in the brain in response to infection, and then neuroinflammation, so immune cells that are recruited into the brain in response to infection. So about 50% of our animals develop this robust CNS infection. And this is shown here. This is taking what's called a sagittal view of the brain. We cut the brain down the midline, and now we're just looking at the side. And here we've gone in to look for viral RNA, so SARS-CoV-2 viral RNA. You can see this a lot of virus in the brains of some of these animals. And if you look at the cell types that are prominently infected, by far neurons are the most prominent cells that are infected by the virus. And that's just shown here. Here's several neurons that are infected by virus. Again, very similar to what I showed you with the human neurons in the dish. You can see virus in the cell body, but also extending down these long processes. Okay. And I need to acknowledge my laboratory, Hema Yuting and Susanna Furman, who are outstanding third-year graduate students in my laboratory that do all the infections. And then a very good friend and colleague, Rob Edwards, who's in the Department of Pathology here, who's done assisted us within looking at the brains and moving forward. In addition, animals develop some neuropathological features in response to SARS-CoV-2 infection that we see in humans, and that is immune cell infiltration. This is a blood vessel in the brain with numerous immune cells that are migrating in. Another blood vessel that's shown here. And then these are the meninges, which is this kind of thin layer that surrounds the brain. But when you have meningitis, that's what that is immune cells are getting into the meninges and infiltrating into the parenchyma. And I also need to acknowledge Bill Young in the Department of Pathology here at UCI that helped us acquire these images. And then finally, this is a beautiful image of the brain of a mouse infected with SARS-CoV-2, which clearly shows two key features. One, we've gone in and done staining for microglia, which are in red. This is looking for a protein expressed on microglia called IBA1. We see massive microgliosis. And then we're also looking at inflammatory monocytes, which are shown in green fluorescence, right? And these are immune cells that come into the brain. And right here, this is a beautiful image that we call the Christmas tree because it looks like a Christmas tree. But you can see this blood vessel here and numerous monocyte macrophages infiltrating into the brain in response to SARS-CoV-2 infection. All right. So the reason I spent the first two parts of my talk talking about this is because I'm building now to what we're really interested in. And so the question is, does infection of transgenic mouse models of Alzheimer's disease with a murine coronavirus affect AD-associated neuropathology? And so we're kind of not a classic Alzheimer's disease pathogenesis laboratory. We're a coronavirus laboratory that now is getting interested into examining how coronaviruses may influence AD pathology, right? And so I don't need to go into great detail here because this audience knows this, but some of the key neuropathological features associated with Alzheimer's include amyloid plaques, neurofibrillary tangles or phosphorylated tau, inflammation, again, immune cells getting into the central nervous system, or activation of resident cells of the CNS. And all three of these collectively work together to form, that culminates in neuronal and synaptic loss, okay? And so, again, for this audience, there are numerous risk factors that are associated with Alzheimer's disease or susceptibility to Alzheimer's disease that include both genetic as well as epigenetic factors, other external factors like head injuries, cardiovascular disease, lifestyle choices, age and gender certainly are important. But I've circled here infections because there's a growing body of literature that has clearly shown that patients with dementia, if they get simply upper respiratory tract infections or other types of infections, it exacerbates some of the clinical features of their disease. And then I use that to dovetail or springboard into the next thing. And the question is, does COVID-19 affect the severity of Alzheimer's disease or dementia? And there's a growing body of research that clearly shows that SARS-CoV-2 infection exacerbates dementia-related clinical symptoms, all right? And you can just go on to PubMed and look at a number of these articles, right? So there's clearly a link between COVID-19 and the severity of dementia or Alzheimer's disease. So what we're interested in then is trying to figure out what are some of the mechanisms by which these occur, and then if we identify this, can we then develop therapeutic strategies to ameliorate this? All right, so I'm kind of going to go back now and just introduce, again, coronaviruses, all right? So in the big picture, I'm sure everybody knows now, coronaviruses are an RNA virus. They have a very large genome, and they're in the beta coronavirus family. And beta coronaviruses include SARS-CoV-1, MERS-CoV, and then, of course, SARS-CoV-2. But importantly, it also includes the virus that we've been looking at for the past 20 years, which is mouse hepatitis virus, or it's a mouse coronavirus, okay? And these beta coronaviruses have a very wide range of hosts, including humans, of course, pigs, cattle, bats, camels. And symptoms associated can be benign to very severe, right? And they can include pneumonia, diarrhea, inflammation of the gut, but importantly, also neurologic disease, neuroinflammation, inflammation or encephalitis, and neurodegeneration. And so that's where we come in. So we can use mouse coronaviruses to study neurologic disease in animals. And so the three main pillars of work that we have going on in our laboratory are to infect Alzheimer's disease mouse models, so transgenic mice engineered to develop Alzheimer's-like disease, infect them with mouse coronavirus and look at the development does this infection accelerate the onset of neuropathology? So in other words, amyloid beta, plaque formation, tau pathology, and inflammation. And these are some of the animal models that we're using, two of which were developed here at UCI. In addition, next month, we're going to start infecting these transgenic mouse models with SARS-CoV-2. All right, so to see if direct infection with the etiologic agent of COVID-19, does that also accelerate neuropathology in these animals? And finally, We're also starting now just getting tissues from patients, Alzheimer's patients that died of COVID-19, in order to see if there are neuropathological changes in the brains of these individuals that's associated with COVID-19 infection. All right, very quickly, mouse coronaviruses. You couldn't tell just looking at them. I can't tell if there's a difference. They look identical to SARS-CoV-2 because all coronaviruses have this crown-like appearance, and that's due to the spike glycoprotein that extends from the surface of the virion. All right, and so that's shown here. The JHM, it's very, it loves to infect and replicate in the central nervous system. It can infect neurons, but other cell types that are infected include astrocytes, oligodendrocytes, and microglia. And this leads again to neuroinflammation and neurodegeneration. And you can see here, we've done this enormous amount of work on coronaviruses, during these mouse coronavirus infection of the brain. And just showing you an example here, we can do an intranasal infection of mice. And then we take the brains out. Here's the brain. We cut it down the midline, and then we can look at the sagittal section shown here. And what's interesting, kind of what we think might be happening with SARS-CoV-2 infection of humans, the olfactory bulb, or the area of the brain that's kind of connected to the nose, we can detect virus in the brain following mouse hepatitis virus very early following infection. But by 7 days post-infection, the virus just goes crazy and replicates throughout the brain. And we've used this as a model to, again, identify mechanisms by which the immune system can control replication as well as participate in disease. Now, importantly, we know that there's a very robust and orchestrated immune response following infection of mice with these coronaviruses. And so shown here is the replication of virus in the brain. It peaks around 5 days post-infection, but then it declines below levels of detection. And that correlates then with the onset of different immune cells, right, that are attracted into the brain following infection with virus, okay? All right, so here are the models that we're currently employing right now for Alzheimer's disease to see if mouse coronavirus infection influenced disease progression in these animals. So we have what's called the triple transgenic mouse, and this is an animal developed by Frank LaFurla, who is now our Dean of Biological Sciences and a recognized leader in the world in the pathogenesis of Alzheimer's disease. We also have human A-beta knock-in mice, which also were developed by Frank and then another professor, Kim Green here. And then 5X FAD mice, which were developed at Northwestern University. And again, we're going to infect these animals and look at tau formation, amyloid beta deposition in the brain, as well as neuroinflammation, okay? And so we actually kind of jumped into this 10 years ago because Frank's been a longtime very good friend and collaborator of mine, and we were talking. I'm like, we should put virus in the brain of your mice. This is obviously way before SARS-CoV-2, and see if this influences neuropathology. And he's like, that's a good idea. Let's do it. So this was done with Michael Tsai, who was an MD PhD student in Frank's lab at the time, and now he's now a neurologist here in the Department of Neurology, and he's set up a clinic on long haulers. So Mike and I are collaborating right now, looking at patients with long hauler symptoms and see if we can identify some of the mechanisms by which this disease continues in these people. But in the triple transgenic mice, we can infect them with our virus. And what we saw is a dramatic increase in phosphorylated tau, or neurofibrillary tangles, right? So viral infection of the brain of these animals dramatically increases tau formation. And this is also correlated with a robust immune cell infiltration. And now, very preliminary studies that we have going on, and this is in collaboration with Kim Green's lab, with a wonderful grad student, Kate Sormus, who's been working with a grad student in my lab, Susanna Furman. We can take these 5X FAD mice, all right? So these are genetically engineered animals, and these animals naturally get amyloid plaque deposition in the brain as they get older. And so our question is, if we infect them with mouse coronavirus, does that accelerate or does it affect the accumulation of these plaques? And indeed it does. The preliminary data shows that if you look in the brains of these animals, and this is 14 days following infection, in the control animals, that is those animals that are not infected with virus, we certainly see plaques because that's what these animals do. They develop plaques, but infection with a neurotropic coronavirus not only increases the density or the number of plaques, but it also increases the overall size of the plaque. So here is a graph showing that in this pilot experiment, we see an increase in the number of plaques in discrete areas of the brains of these 5X FAD mice infected with virus, but also we see an increase in the plaque size. And what's intriguing here is if you look by immunostaining, again, these are sagittal sections of the brains. This is a control or uninfected 5X FAD animal. You can certainly see some plaques in the cortex here, But this is a JHM infected animal or mouse coronavirus infected animal. And here you can see two key things. One, if you look in the cortex, there are a lot more plaques and they're a lot bigger, which is consistent with what I showed you before. But we're also seeing a very robust immune cell response, which is something we anticipated because the immune system is going to respond to a viral infection of the brain. But you see this rapid accumulation of these monocytes, all right? And these are immune cells in the blood that then go to the brain in response to infection. And so we're very curious if these monocytes that are getting into the brain, do they have any role in perhaps accelerating or augmenting some of the disease that we're seeing? So to conclude, using these different transgenic mice, and by the way, I didn't touch on the human A-beta knock-in mice, but those studies are ongoing right now. But using these different transgenic mouse models of Alzheimer's, we've been able to show that infection of the CNS with a mouse coronavirus increases AD pathology that's associated with what you see in patients with Alzheimer's disease, and specifically phosphorylated tau and amyloid beta accumulation. And this increase in pathology in these brains was associated with immune cell infiltration, suggesting a potential role for these cells in enhancing disease. And so right now, what we're going to be doing is attempting to specifically block targeted populations of immune cells from getting into the brain and seeing if that ameliorates or reduces or maybe increases the severity of amyloid plaque deposition or AD pathology. And then finally, the experiments we're really excited to start, which we'll be doing next month, is to see if AD, infection of a transgenic AD mouse models with SARS-CoV-2 accelerates the neuropathology. But then what we're also building into this study is looking at memory consolidation and cognition and seeing if this virus infection affects these pathways in these animals. And then finally, we're getting brains from patients with Alzheimer's disease that succumb to COVID-19 to determine, can we see if there's increased penetrance of virus in the brains of these individuals? And is there an increase in neuropathology? And then finally, do we see an increase in immune cell infiltration? I'd like to conclude by thanking my wonderful collaborators here at UC Irvine, specifically Susanna Furman, who's a graduate student in my laboratory, who's done all the work that I've shown you in collaboration with Kate, who's a graduate student in Kim Green's lab. I'd also like to acknowledge and thank Dr. Lindsay Hostfield, who's a postdoctoral fellow in Kim's lab that's also been instrumental in these studies, as well as research funding. And I will conclude here, and I look forward to taking any questions. Thank you.
Okay, we are joined by my esteemed colleague, Dr. Tom Lane, live now. Tom, thanks so much for a fantastic talk. The questions have literally been pouring in during your talk.
Okay.
And thanks for being here live with us. So to get us started right out of the chat, is it known whether neurological symptoms are a direct result of the virus or due to collateral damage, perhaps from an active immune response, for example?
Right. Very good question that a lot of people within the field of COVID research are continuing to ask. And so we know that the virus does have CNS penetrance, right? That it can get into the central nervous system, but it's not, when you do postmortem analysis of tissue from patients that passed away from COVID, there's just not a lot of virus there. It's in discrete areas. It's primarily infecting neurons. So, I mean, it's certainly some of the neurologic consequences, and perhaps this is true more for people with post-acute sequelae of COVID or long haulers. There might be more virus there. That story's that's still ongoing. But, I'll tell you, Josh, I think a lot of it is going to be due to this robust acute inflammatory response that occurs in the periphery. And as well as I that, inflammatory cytokines and chemokines can cross the blood-brain barrier and alter the microenvironment in the central nervous system. So these are some of the areas that we're looking at to address in animal models.
Yeah, and it's so exciting to have you back at UCI and bring this expertise with the expertise that was already here doing animal research in Alzheimer's disease. I think you guys are going to make incredible contributions as we continue to study this.
Well, thank you.
Another question from our audience, what about other viruses? Do other viruses affect Alzheimer's disease? The question was specifically about the flu virus, which we know is incredibly common, but what do you think about other viruses?
Well, there's a body of literature that indicates that people with dementia that get upper respiratory tract infections, so for example, from influenza, you do see exacerbations of some of the clinical symptoms associated with the disease. And I think that's probably the result of the inflammatory response, right? But better than I, the whole herpes simplex virus story that was very prevalent several years ago, and that's waned. There's an interesting literature that argues that treatment with antiviral drugs, or like acyclovir and maybe other things that block herpes replication, has beneficial effects. I think that needs to be looked at in a little more detail. There are a lot of neurotropic viruses out there that are often benign, and then if you have a genetic predisposition for developing dementia or Alzheimer's, I think we need to start looking in a lot more detail about what these viruses are really doing and how they affect the neuropathology.
Yeah, thank you. I totally agree. I think that is a fascinating story, and it produced really important hypotheses that need further work. But of course, any study that suggests a link between treatments or illnesses really requires more prospective research before we can even consider thinking about impacting how patients should live their lives.
Exactly.
You kind of mentioned genetics, and there's a question here from one of our audience members about whether ApoE4 influences susceptibility to COVID and its influence on Alzheimer's severity. I know ApoE is tough to study in mice, but do you have any thoughts on that one?
These are some of the directions that we're heading, right? We do know that there's an increase in ACE2 expression in the brains of patients with Alzheimer's disease, right? And so, again, that's one of the receptors that SARS-CoV-2 employs to gain access into host cells. Now, whether that is due to other genetic manifestations that lead to Alzheimer's disease, we don't know. But what we're trying to do now, Josh, is we're starting next week infecting transgenic mice with SARS-CoV-2, and what we can do is start doing some of the genetic manipulations with ApoE, TREM2, et cetera, and see how that alters either the penetrance of the virus into the brains of these animals and then ensuing neuropathology. So that's a very good question. It's a very complicated question, but it's very relevant.
Yeah, and I don't mind telling our audience, you and I were chatting during your talk a bit, and the next question from an audience I think is timely for you. Do you think the main differences in neuroimmune response due to coronavirus infection leading to increased amyloid burden versus that caused by something else, which can lead to reduced amyloid burden?
So let me repeat the question to make sure I understand it. So that the viral, with the mouse coronavirus, the increase in pathology that we're seeing is due to the viral replication, but then perhaps the inflammatory response can also alter the pathology one way or the other. And so what we were talking about right now, in the early stages of infection, we do see this trend towards an increase in amyloid pathology in the brain. But now, as I was telling you, 3 days ago, we just got these really compelling data. As the disease progresses in these animals, we see a greater accumulation of inflammatory immune cells, particularly macrophages, and we start seeing a reduction in the plaque morphology or plaque burden. So that argues that maybe these cells are coming in and eating up plaque. So there's a lot of really intriguing questions that we're getting into right now. And so it's complicated, but it's very exciting at the same time.
Great. Tom, another question I'm sure that's on everybody's mind. What about the variants? Are all the strains of coronavirus going to be the same when it comes to impact on Alzheimer's disease?
We don't know. It's an extraordinarily important question. And, the one thing about coronavirus is that I'm really keeping an eye on, we've been doing coronavirus research for over 25 years, is they're very large RNA viruses, right? And they're not segmented. That's a good thing, but they can recombine at a very high rate. And what that means is most of the times you just make bad viruses. They're not infectious. But what I'm worried about is, over time, are we going to see a virus that does have greater CNS penetrance, right? That can really get into the central nervous system and cause a lot of damage. And so I think those are the things that I'm looking at right now and what I'm intrigued about.
We have only a bit more time. Another question. could you offer your opinions about whether someone infected with COVID who's asymptomatic and may not ever really know that they're asymptomatic still have long hauler syndrome or develop neurological symptoms down the road?
Yeah, that's certainly the case. That's why long haulers is so, enigmatic, if you will. You can have, for example, as the question relates to, you completely asymptomatic, no loss of smell or taste, and then 6 months later you start developing loss of smell and taste, for example, right? So why is that? And so there's some interesting literature that's coming out from UC San Francisco that argues that there might be some misguided attack during the course of your immune system combating COVID, that you're generating antibodies that get into the central nervous system and attack unique proteins in the brain, in the CNS. Now, is that an underlying factor that is contributing to the development of long hauler syndrome? We don't know. Is there a greater penetrance of the virus into discrete anatomic areas in the central nervous system? Again, we don't know. So we're starting right now with Dr. Mike Tsai, who I think you probably know. He's got a long haulers clinic here at UC Irvine, and then we're also reaching out to a group up at UCLA to start looking at people like the CSF and cerebral spinal fluid from patients with long haulers and start looking at this in more detail. And then also, we're trying to figure out how to do memory consolidation experiments in mice that are infected with SARS-CoV-2, which that's going to be tough, but we can do it.
Yeah, we've only got about 20 seconds. There was one last question about whether people with Down syndrome are increased in their vulnerability to COVID-19. Obviously, it's a big portion of our community for UCI Mind.
Yeah. There seems to be that seems to be the case, and there are probably a lot of different sociological aspects associated with that, and particularly, there also might be biological effects. We are getting right now, next week, we're getting post-mortem brain samples from Down syndrome individuals that passed away from COVID, just to see, do they have a higher increase of brain penetrance by the virus compared to unaffected individuals. So it's another really good question.
Great. Tom, this has been fantastic. Thank you so much. We really appreciate your talk, your being here, and your being here. So thanks again.
Well, thank you so much for the opportunity, Josh. It's you're one of the main reasons why I wanted to come back and work with people like you. So thank you very much.
Thanks. For our audience, we're gonna take about a 7-minute break full of important information that we hope you'll stay tuned and listen to.
With United Healthcare Medicare Advantage plans, there's more to take advantage of. Get a checkup here and have a wellness visit come to you there. Pick up your prescriptions here and get free delivery there. Stay active with free home workout videos here and a free gym membership there. See a doctor in the morning here or talk to a nurse in the moonlight there. That's more to take advantage of. Here, there, and everywhere. United Healthcare Medicare Advantage plans. Take advantage everywhere.
You've spent a lifetime making memories. We invite you to make more. At Belmont Village, residents find the perfect balance of exceptional living and exceptional support. A lifestyle to suit you, and for couples who need different levels of care. an experience designed by experts in hospitality, dining, architecture, and enrichment. It's not just the life you need, it's the life you've earned. Belmont Village Senior Living, the community built for life. You've spent a lifetime making memories. We invite you to make more. At Belmont Village, residents find the perfect balance of exceptional living and exceptional support. A lifestyle to suit you, and for couples who need different levels of care. An experience designed by experts in hospitality, dining, architecture, and enrichment. It's not just the life you need, it's the life you've earned. Belmont Village Senior Living, the community built for life.
I'm Diane Mondini. I'm president of Caring Companions at Home. I got involved in Alzheimer's OC back in the late 90s when my good friend Julie David served on the board and encouraged me to help with the galas. Because of my business and dealing with seniors for the last 30-odd years, I have seen how this disease has taken its toll on many of my clients and many of my clients' family members. It's very hard seeing them suffer and wondering what you can possibly do to help them. That's why I have a passion for trying to find a cure for this disease, or helping in any way I can.
My name is Gloria Barr. I have always been independent and thought myself quite capable. But now that I'm 92, I find that it's not as easy. I heard a lecture here at the clubhouse and I signed up with Independence at Home. They have been terrific.
When I came to see Gloria, she was having some issues to open her pill bottles. I was just happy I was able to provide that to her and avoid any injuries.
I can open bottles now like Atlas. It's just a wonderful outreach to the community that I certainly would recommend to anyone.
Okay, welcome back. I hope you enjoyed those messages from our sponsors and partners. You are in for a real treat now. Our next talk is from my friend and colleague, Dr. Amy Kind from the University of Wisconsin. Dr. Kind is the director of the Center for Health Disparities Research at UW, and she's also the leader of the Care Research Core for the Wisconsin Alzheimer's Disease Research Center. Dr. Kind developed the Area Deprivation Index and then took on one of the greatest data democratization efforts known to our field. She published this ADI in the New England Journal of Medicine and has made it freely available to investigators nation and worldwide. And the results of that effort are hugely impactful. She and others have published papers about the importance of neighborhood in a variety of health outcomes, now including Alzheimer's disease. The neighborhood in which you live has an impact on the likelihood that you will get plaques and tangles, the size of your brain, and even your cognitive performance over time. We are thrilled that she has chosen to work with us at UCI. We're part of one of her new very large grants to study the ADI's role in aging and Alzheimer's disease. And I'm grateful to her for collaborating with our group as well. So please enjoy this lecture.
Hello, everybody. My name is Amy Kind. I'm a professor of medicine at the University of Wisconsin School of Medicine and Public Health. I direct our UW Center for Health Disparities Research, and I lead the CARE Research Corps in the Wisconsin Alzheimer's Disease Research Center. I am so incredibly pleased to be able to be with you virtually today and would like to thank Dr. Josh Grill and the UCI Mind team for the opportunity to present. So today we are going to be talking about geographic disparities and how location could potentially affect brain health. There'll be an opportunity for questions near the end. So here are my disclosures. So disparities in Alzheimer's disease and related dementias are all too common. We have seen repeatedly that dementia due to ADRD disproportionately impacts certain populations. For example, rigorous population-based studies from England and China have demonstrated connections between poverty and brain health metrics, as you see here. Additionally, studies from the United States have repeatedly shown linkages between race, ethnicity, and brain health, with African Americans and American Indian or Alaska Natives being at greatest risk. So brain health is not equally distributed within our world. Unfortunately, effective interventions to eliminate AD disparities are not yet available. And as a field, we need solutions, and we need them soon as possible. And to do so, we need to better understand the mechanisms that drive disparities in ADRD. When we can understand the mechanisms, we can develop better opportunities for therapies, cures, and interventions. However, as we do our research, it's also incumbent upon our field that we move forward in a manner that is aligned towards real-world action. Research aligned towards action may include studies that help forward opportunities for new treatments, new clinical intervention, and new social policy. This is really an ethical responsibility of every researcher in our field, but unfortunately, not all research is aligned towards action. There are projects that may provide descriptive understandings, but don't necessarily provide a path forward toward real-world change. So we all, as a community, as a research field, must focus on factors that are modifiable. So in today's talk, I'll be discussing some of the latest theories of the factors that are believed to drive brain health disparities, with a particular focus on social contextual factors. We will then discuss one approach to align such research towards real-world action, highlighting some of the latest cutting-edge findings within the field. Finally, we will discuss the power and the importance of open data. I will share some opportunities for us all to join together in alignment towards improved real-world change by targeting modifiable factors. So first of all, let's ask the question, why do we think disparities in brain health occur? The National Institutes of Health and the National Institute on Aging's answer to this question is provided by their mechanistic health disparities theory that you see here. In this model, health disparities are driven by a complex array of environmental, sociocultural, behavioral, and biological factors that interact across the life course to produce health and disease. In viewing this model, it becomes clear that as a field, we need an in-depth understanding of the mechanistic interconnections among social and biological factors. Such an understanding would help us to know more about why disparities happen, to get to the roots of their mechanisms. Many of the factors in these models are external to the individual. That is, they're outside of a single person. These are environmental or social factors that we need to understand more about because they play an important role in disease. These types of factors are titled or are called the exposome. So an exposome is a measure of all of the exposures of an individual in a lifetime and how these exposures relate to health. Such linkages could form the foundation for new foundational disease mechanisms, new therapies, precision medicine approaches, and other interventions. But we are just beginning to learn about these areas. As we note the complex interconnections of these different factors, it also becomes clear that not all of the factors that we study and that we think drive health disparities are modifiable or actionable. In this model, as in countless others within the health disparities field, the social determinants of health have a particular place of prominence. Social determinants are noted prominently because they typically are modifiable and can serve as potential levers for action. So we know that the social determinants of health have a profound impact on the life, health, and well-being of all individuals. And the scientific field in general has known about this for many, many decades. The U.S. Centers for Disease Control defines social determinants as the conditions in the environments in which people are born, live, work, play, worship, and age that impact a wide array of health, functioning, quality of life outcomes, and risks. Social determinants have been demonstrated as having a much larger impact on population health than even clinical care, and have been shown to impact the very foundations of human physiology. Differences in the allocation of social determinants often reflect political, social, and structural practices of inequity that are driven by unfair policies based on race, income, religion, immigration status, sexual or gender identity, geography, or other characteristics. Differences in social determinants can reflect legacies of historical injustice in a particular region or country. We can also say that social determinants exist at both the individual level as well as the contextual level. Individual level social determinants of health include an individual's education, their occupation, and their income. Excellent research in the ADRD field by researchers such as Dr. Rachel Whitmer, Dr. Jennifer Manley, Dr. Lisa Barnes, and many others have demonstrated associations between individual level social determinants and numerous markers of brain function and health. Contextual level social determinants include factors such as neighborhood disadvantage and the built environment. Decades of social science research have demonstrated that these contextual level determinants exert profound impacts on human health. Yet, unlike the individual level determinants, the study of these contextual factors in relation to brain health is still quite early. There's much to be done. Nonetheless, as you will see shortly, some of the early findings that are coming out of this area are extremely exciting. So one might ask, why should we focus on the context? Do neighborhood social determinants impact health over and above the impacts of those individual level factors that we just discussed? And how might health differ for an individual who lives in a typically wealthy context as compared to a highly disadvantaged context? We know from other fields of study that context, and most specifically the geographically discrete neighborhood level disadvantage, so very specific context measures, are strongly associated with health independent of those individual level social determinants. The context in some cases seems to have an even larger effect on health for certain conditions than the individual level social determinants. An excellent example of this can be seen in the Moving to Opportunity study, which was a natural policy experiment that occurred in the 1990s US. During this time, the Clinton administration had a goal to eliminate poverty. One way that they hoped to do this was by establishing a novel program in which poor families in certain US cities could enter a lottery for a specialized housing voucher, which would allow them to move to a slightly more wealthy area. It wasn't necessarily a highly wealthy or a very rich neighborhood, but it was slightly more wealthy than the typical Section 8 neighborhoods. Many families in these eligible areas applied, and some of these families were lucky enough to receive the vouchers. Social science researchers at the University of Chicago realized that this was a natural random experiment. They started enrolling and tracking the families, and after decades of follow-up, it was found that this grand experiment did not eliminate poverty, which was the hope. Individual socioeconomic social determinants, including income, did not change. However, and somewhat unexpectedly, researchers found that health of the individuals of these families who moved into the less disadvantaged neighborhoods was markedly improved. Cardiovascular disease, diabetes, mental health, all seem to have been impacted. And this finding, supporting the power of the neighborhood context, has been replicated across many disease types and studies. Cancer outcomes, cardiovascular health, obesity, surgical survival, frailty, and more. They all link more strongly to the neighborhood disadvantage than to individual level measures. Also critically, neighborhood disadvantage is modifiable. It's actionable in the real world. Context can be changed through policy, through clinical intervention, and through resource allocation. It is this combination of impact and modifiability that makes a focus on the context so very imperative in ADRD research. Measures of neighborhood disadvantage also reflect legacies of structural inequity in many regions of the world. In the United States, metrics of neighborhood disadvantage reflect a legacy of structural racism in many areas, particularly urban areas. This is a historical map of one city that embraced a process called redlining. Redlining is the systematic denial of various services by federal government agencies, local governments, or the private sector that ultimately disadvantaged poor and minority communities. So areas in red on this map did not have the same access to financial resources as the areas in blue. So neighborhoods who might have otherwise thrived were systematically denied access to the loans and financing essential to infrastructure investment. The legacy of these inequities echo into the present day. So we might next ask, can we reliably measure neighborhood disadvantage? And the answer is a resounding yes, and comes to us from the field of health policy. Metrics of neighborhood disadvantage are already developed and often leveraged in health policy infrastructures of countries throughout the world. They are typically used to inform health system resource allocation, particularly in countries outside of the United States. So they're quantifiable. They use measures of social determinants, again, income, education, housing, quality, and employment in a very discrete geographic area. So usually a population-based area of typically 500 to 1,500 persons. This would be equivalent to the United States Census Block Group. These metrics are robust in that they're available across different countries, they're privacy compliant in that they can be shared across the internet, and they're very actionable in the real world. You can guide outreach through these maps and think about targeting areas in greatest need. So they can be used for not only targeting, but also determining eligibility for certain types of advanced program supports, and they can be used for statistical adjustment. The first of these indices was developed in the 1980s by a Dr. Brian Jarman from the UK. Dr. Jarman was a leader in the scientific organizations of the UK at that time, the British Medical Society, and decided that he wanted to somehow have the ability to attract more physicians or GPs into areas of the UK that were highly disadvantaged. So he created this metric based on social determinants of health at a very discrete geographic level, the Jarman Index, and was able to rank neighborhoods throughout the UK on this index to determine which areas were the most disadvantaged. Using this, he was able to convince the Margaret Thatcher administration to change the way they paid physicians at the time in order to increase payments to physicians that would practice within these most disadvantaged areas. And these policies were effective. They were able to attract more doctors to these areas of greatest need. So our team was very interested in harnessing a neighborhood disadvantage metric for the US. As I mentioned, these metrics have been primarily used in Europe and outside of the United States, but we thought they had a lot of potential for US-based use. So we've worked over the past decade to refine a metric called the Area Deprivation Index, or ADI, which is a metric similar to the Jarman Index, but for use in the United States. The ADI was originally created in the 1980s by the Health Resources and Services Administration, a small branch of the US federal government. But it was employed at the county level, which was not in alignment with the European indices. It did include measures of education, employment, housing quality, and poverty, but it required quite a bit of update for modern use in that the data that it drew from no longer existed. It uses census data that needed to be updated, and it needed to be refined down to this much more precise level of geography. So our team took up those challenges almost a decade ago, and we've been working on that ever since. We refined the metric down to the census block group, which is again about a neighborhood population based about 1,500 persons. In an urban area, it can be very, very tiny, and within a rural area, these block groups can be very geographically large to meet that population threshold. This allowed us to much more precisely measure exposures to disadvantaged contexts across the US. So for example, on the map on this slide, the entire area that is being shown here is one county. This is Milwaukee County. It's one of the most disadvantaged counties within the state of Wisconsin. And if you were to look at this county based on simply county-based rankings, which again would be this whole area, this entire county would be labeled as highly disadvantaged. However, when we use this much more precise, geographically precise metric, what we see is that Milwaukee County has an inner city core of bright red. Bright red on this scale is the most disadvantaged areas, surrounded by a ring of bright blue, and blue on the scales and maps that I'll show you are the least disadvantaged or the most wealthy contexts. So we see that the county is actually has an extremely large range of neighborhoods that range from highly, highly disadvantaged, profoundly disadvantaged, to some of the most wealthy neighborhoods within the state of Wisconsin. And so the precision of our metrics allow us to move forward in some of the ways that I'll show you later on in the talk. So we might next ask, what are the characteristics of the most highly disadvantaged neighborhoods in the US? So these red areas on these maps, these heat maps that show disadvantaged neighborhoods and are produced by our team. So in the United States, highly disadvantaged neighborhoods are most often found in urban core and highly rural areas. Furthermore, the racial proportions differ by neighborhood disadvantage in the US. From a nationally representative study of US older adults, 90% of those living in the least disadvantaged neighborhoods self-identified with the racial category of white, and 7% self-identified with the racial category of Black. However, those proportions differ in the most disadvantaged US neighborhoods. 58% of individuals residing in the most disadvantaged US neighborhoods self-identified with the race category of white, and 33% self-identified as Black. 9% self-identified with another racial category. So we have been collaborating widely in sharing data openly with many independent groups to understand more about how the social exposome, in this case, neighborhood disadvantage, might be linked to brain health. By leveraging and using metrics such as the ADI, we can perform AD-RD research that becomes more quickly actionable and harmonizable across a wide swath of domains. In this study, we recruited 951 cognitively unimpaired research participants, and their residential address was geocoded, linked to neighborhood disadvantage by ADI. And then we used MRI measures of hippocampal and total brain tissue volume to learn more about their brain health. And the hippocampus in particular is an area of interest in Alzheimer's disease. We found that living in a highly disadvantaged neighborhood by ADI was associated with lower hippocampal and total brain volumes by MRI, independent of individual factors, so independent of the individual socioeconomics. So we extended this study by following a longitudinal cohort over 10 years. And in this 10-year longitudinal study, again, of cognitively unimpaired adults, we found that living in the most highly disadvantaged neighborhoods was associated with accelerated degeneration, that is cortical thinning, in AD-affected regions and more cognitive decline. So these are individuals compared to themselves. And for those that were living in the most disadvantaged contexts in this particular study, they had accelerated degeneration of their brain. In addition, differences in brain structure have been seen as early as age 2 in children who live in highly disadvantaged contexts. In study that is being led by Dr. Ramphal from the New York City area, he has found multiple differences, and it is quite notable and heartbreaking. But what about Alzheimer's disease neuropathology? Neuropathology is the gold standard in diagnosis of Alzheimer's disease. So we were interested in linking these metrics of neighborhood disadvantage to neuropathology. So we collaborated with the University of California, San Diego, and along with our brain bank at the University of Wisconsin, and assessed 453 decedents who had donated their brains to one of these 2 brain banks. We were partnered with Dr. Robert Rissman at UCSD, who leads the brain bank there. These were individuals who had donated their brains between the years of 1993 and 2016, and no social factor characterization was available. So these were individuals that had tissues that were exquisitely characterized, but very little was known about their social characteristics. We took the residential address at the time of their death and geocoded that and linked it to neighborhood disadvantage, again, by these ADI metrics that I showed you, and took neuropathology features that had already been reported up to the National Alzheimer's Coordinating Center and from the existing autopsy reports on these individuals. So what did we find? We found that living in the most disadvantaged neighborhood decile was associated with an increased odds of Alzheimer's disease neuropathology. This was a small study. It needs to be expanded, but this line of research is exceptionally exciting to me because it is now possible to append rigorous measures of social context to existing tissue that has not otherwise been socially characterized. Additionally, our team has developed methods by which we can obtain measures of context across the full life course of these decedents, from birth to the point of brain donation. We do this through residential history, which allows us to perform geospatial targeting of exposure data across the life course. We create a list of addresses from the point of birth to the point of death. And we can do this using survey in living cohorts, but also using genealogical and historical methods for individuals who have passed away and have donated their brains to science. Using these techniques, 73% of all person years are discoverable. So it's very, very exciting, and it gives us another way to understand more about how the social exposome may be linked to brain health and brain function. So this technique opens the door to something called social biological characterization, and it allows us to apply this to existing tissue and biospecimen repositories across the United States. Brain donation is an incredibly precious gift to our field, and this type of research allows us to expand the impact of that gift, even if that donation was made decades before. So we can also say that residing in a highly disadvantaged neighborhood is linked to multiple other health outcomes and multiple other factors that are important in our area of Alzheimer's disease. So for example, epigenetic age acceleration as measured by methylation-based markers of aging was found by Lawrence et al. For individuals who live in more highly disadvantaged neighborhoods. Living in a highly disadvantaged neighborhood is associated with a higher risk of postoperative delirium, with cognitive function and decline, with difficulty accessing care, including later dementia diagnoses and less comprehensive diagnostic evaluations, and greater challenges navigating dementia care and supports for the caregivers who live in these areas as well, and certainly many, many other factors. In addition, we can say that the pathways to brain health seem to be strongly associated with living in a highly disadvantaged neighborhood. So on this slide, we see the level of cholesterol control amongst individuals who are participating in Medicare Advantage programs across the United States. And higher is better on this graph. And what we first see is that individuals who self-identify as Black or African American across this entire graph have the lowest percent control, so have the most difficulty achieving cholesterol control for a whole host of reasons. So the best control as well can be seen for individuals who live in the least disadvantaged neighborhoods. So the least disadvantaged neighborhoods on this graph are all the way to the number 1, whereas individuals who live in the most disadvantaged neighborhoods over here up to the 20 on the ADI ventile scale show the worst cholesterol control. So not only do we see a race and ethnicity disparity here, but we also see a disparity by where individuals live. So individuals, African Americans who live in the least disadvantaged neighborhoods tend to have much better cholesterol control than individuals who identify as African American who live in the most disadvantaged neighborhoods. So again, this allows us to unpack some of these mechanistic features around health disparities. And cholesterol control becomes so important for brain health. If we control our cholesterol, this is one of the risk factors that can lead to heart disease and other vascular events, which of course are something that we all want to avoid. We can also say that persons residing in the most disadvantaged neighborhoods are often poorly represented in research. I would argue that neighborhood disadvantage should be collected as a standard demographic characteristic in all research protocols. And what we tend to see, regardless of the protocol that we're looking at, is they tend to overrepresent individuals who live in extremely wealthy contexts and underrepresent individuals who live in the most disadvantaged contexts, just like the graph on this slide. We need research on best practices for recruitment and retention of individuals who reside in disadvantaged neighborhoods, and we need it soon. And wonderfully, as a first step towards understanding more on this topic, Dr. Josh Grill and his UCI team right here in your site geocoded their C2C research registry and linked those findings to ADI. So the blue bars on this slide represent the proportion of all individuals in the catchment area by ADI. Again, low numbers mean more wealth, high numbers mean less wealthy neighborhoods. The red bars are the proportion of individuals recruited to the C2C research registry by ADI. So first of all, as you can see, fewer registry participants originate from the most disadvantaged neighborhoods. However, when Dr. Grill and his team asked individuals, queried individuals, they found that regardless of the individual's level of neighborhood disadvantage, residential neighborhood disadvantage, all individuals had a similar willingness to participate in research studies. So efforts to increase representation of participants from disadvantaged neighborhoods in registries could be an important first step towards increasing the generalizability of clinical research. So to realize such change, though, we must embrace open science. Open science is the movement to make scientific research and its dissemination accessible to all, not just to highly educated PhDs, but to all persons in a society. This act of open science is termed data democratization and is one pathway that can empower research towards becoming actionable. This is critically important because many of the persons and structures in control of the levers for social, policy, financial, and clinical systems change are not directly part of the ADRD research community or of any research community. Furthermore, many of the individuals in the communities that are impacted by ADRD are not directly part of the research community. Yet, these are the diverse populations that can stimulate, advance, and sustain real-world change. These are the persons that we must include in our science. So our group has embraced this principle of open science, and we have done so through this tool called the Neighborhood Atlas, which you can see the address for here. You're welcome to take a look. It is a data democratization tool that allows free and open access to all of the data that I've shown you today in terms of neighborhood disadvantage. You can do customized mapping, very similar to smartphone apps that you may each have on your phones if you have a smartphone. And to date, it's had over 250,000 views, with the mapping options being the most popular. Additionally, it allows options for data downloads of over 79 million data points. These data can be used in health, policy, and research and governmental activities, and they have been downloaded by all of these stakeholders. So this is the Neighborhood Atlas. This is what it looks like on the homepage. You can choose your state of interest. This is Florida, for instance, and you see these heat map-based formats of highly disadvantaged areas in Florida as compared to the blue, more wealthy regions. You can zoom in and see landmarks such as streets. This becomes very important for community-based outreach. And so different groups have been using these metrics and using them widely. So we have found that embracing data democratization has proven to be a very powerful step in catalyzing real-world change in research and advancement. And in just a handful of years, hundreds of groups have published using our Atlas data. So just a few examples here, as I mentioned, our embracing of open data methods have allowed others to forward the field in important ways that we could never have imagined. This is evidence of a community aligned towards action. And excitingly, simultaneously and synergistically to these research publications, many policymakers, health systems, and community members have also used these very same Atlas data to trigger real-world change. So we've had groups as diverse as the Maryland Health Services and Cost Review Commission that does a lot of work in hospital payments across Maryland, to the state of Pennsylvania, who incorporated the ADI in its official COVID treatment allocation policies. We've had states across the country who have used the ADI and the maps on the Neighborhood Atlas to decide where COVID vaccine allocations would be targeted. So many, many uses. Certainly, the opportunity to intervene on neighborhood disadvantage and to study this challenge has never been better. And for us, by embracing open data and data democratization, our research has grown and expanded. I'm extremely pleased to announce that our latest action-oriented study was just funded by the National Institutes on Aging. This $28.5 million neighborhood study is a novel collaborative multi-site initiative to examine life course exposome on AD-specific outcomes across 22 ADRCs, those are Alzheimer's disease research centers, including 52 key personnel, and led by myself and Dr. Barbara Benlin, a professor here at the School of Medicine and Public Health at Wisconsin. This study will include over 9,000 ADRC brain bank donated decedents and approximately 7,800 clinical core participants with a rich array of imaging studies. The neighborhood study will result in new opportunities for action-oriented scientific inquiry and open science. It will allow us to perform life course assessments of existing brain tissues. In so doing, we will have the opportunity to understand more about the dosage and timing of disadvantage exposure across the life course and how that might be related to brain health and ADRD. It will also enable a diverse consortium of researchers to gather in alignment towards real-world action. The neighborhood study will set the stage for a multitude of future life course exposome research and intervention, and future expansions are already being planned. So stay tuned for more on this study in the coming years, and we're pleased that UCI is part of this group. So in conclusion, I'd like to end with a few thoughts. I would argue that whenever possible, research should be aligned towards action and real-world intervention. This is something that we all have to keep in the front of our minds as researchers, but hearing the voices of participants becomes very important in this as well. So be sure to tell your researchers about what matters to you and what types of actions need to be taken as well. Social determinants of health, including neighborhood disadvantage, drive many health disparities and often reflect a society's legacy of structural inequity. Neighborhood disadvantage should be collected as a standard demographic characteristic in all research protocols, and we continue to advocate for this across the research community as well as beyond. And then finally, open science, data democratization really should be embraced as a step towards real-world change, because all of us have a role in mitigating brain health disparities. We can all perform this outreach as long as we have access to the tools that we need. And importantly, it takes an entire community to result in some of the changes that we hope to see in our society towards improved brain health. So finally, the Neighborhood Atlas is open to all. Here again is the website if you want to take a look. For those of you interested in metrics for other countries, many ADI type metrics exist. For other countries, just reach out to your health policy colleagues. And certainly I can answer more questions in that in our Q&A session. So finally, our work would not be possible without a large, talented team of individuals dedicated to eliminating health disparities and research participants who volunteer to forward our field. Thank you to all of you. And thank you to the National Institutes on Aging for the opportunity to do this work. And especially thank you to Dr. Josh Grill and the UCI Mind team for the opportunity to present today's talk. It has been an honor. So thank you, and I'll look forward to your questions.
Okay, we are now joined by Dr. Kind, straight from Wisconsin. Thank you for being here, Dr. Kind.
Thrilled to be here. Thanks for inviting me, Dr. Grill.
So happy to have you. I'm going to dive right into questions. The first one comes from a UCI Mind investigator, Dr. Chris Salazar. He's asking, is there a critical period during life when residence might matter most for some of these outcomes later in life?
Oh, it's such a great question. Thank you for asking that, Dr. Salazar. So we are very interested in this writ large as a field, thinking about how someone's exposure across their entire life course from birth or cradle to grave ultimately impacts their health and well-being. How does exposures in early life impact brain development, and does this lead to differences in cognitive reserve, differences in the ability to receive some sort of challenge to the brain and yet overcome that challenge? Could this be something that we tap into as a field in order to make our entire population more resistant to neurodegenerative diseases like Alzheimer's disease? And so our field is just beginning to start examining some of these ideas, looking at life course assessments, both from starting with the pediatric population and following them forward, or using a technique called retrospective assessment, in which we start with information that's been gathered already and move backwards in time using historical methods to add more social information about the individuals who have already been well characterized, having their tissues well characterized or their images well characterized. What we can say right now is that we can say that individuals who live in highly disadvantaged contexts when they're very, very young have very different brain structures than their comparison groups, than other children who live in less disadvantaged context. And trying to understand more about why that might happen and what kinds of impacts such differences could have later on in life is an area of active inquiry. It's one of those really unique areas in which pediatricians, doctors for children, can come together with doctors for older adults and researchers, aging researchers, and ask questions in completely new ways. So, so lots to be learned yet, but lots of tantalizing information.
Great. One of our local KUCI radio hosts, Claudia Shambaugh, asks, what about intergenerational effects? Does where your parents lived have an impact on your brain health or any other aspects of which things might be inherited?
That's also a fascinating question. This idea of intergenerational transmission of health, wellness, and disease is something that has been looked at by geneticists over time, particularly for cancer risk, for some of the inherited disability, challenges with the heart, for instance, those types of risks. But it hasn't been examined in depth for neurodegenerative diseases like Alzheimer's disease. It's a great question, though, because we know from social science literature that where you grow up sometimes has a big impact on where your parents live. Obviously, it's a family unit. And so the children, not in all cases, but in many cases, are living with their parents within the same type of neighborhood context and constructs. So does some of this environmental exposure, does it interplay with our fundamental biology in such a way to produce disease? And how much of this, of any intergenerational transmission of wellness and disease, if there is any, is due to biology and genetics, or is it due to the environment and the shared environment over time and the different types of exposures? It gets to some of the most fundamental questions that are in front of the scientific field as a whole, but certainly are greatly important for our Alzheimer's disease and neurodegenerative fields as well, and dementia. So no answers as of yet, but lots of researchers continue to look at those questions and try to understand more. Certainly, once we find more, it gives us more of an idea of how to respond. How do we fix health disparities? How can we move our society forward as an entire population to help all of us achieve better health? And understanding whether those mechanisms lie in the environment, lie at the cellular level, or a combination of the two, really help us move forward and can be thought of as another branch of precision medicine.
We're quickly running out of time, I'm afraid, but another great question. How do you, what are your thoughts on the recent findings as it relates to air pollution as a risk factor for late life cognitive problems? And if you will, corroborating, the neighborhood aspect of this?
Yeah, those are some very interesting findings, and the associations do seem to be strong with pollution, fine particulate air pollution, and some cognitive outcomes, as well as other types of outcomes that are of import to neurodegenerative diseases. What I can say is that for individuals who live in highly disadvantaged contexts, they are much more likely to experience high pollutions, high levels of pollution. And actually, we know this from many of our environmental scientists worldwide, in that the places that pollution tends to congregate, so the places that air pollution, water pollution, land pollution, those tend to echo the same types of areas in which individuals who are poor, disadvantaged, do not have a lot of agency or power politically tend to live. And those also tend to be the areas that are highly disadvantaged when you look at metrics, multidimensional metrics like the ADI or other variants of those types of metrics in other countries. So there's a lot of interlocking potential overlap there. We will have to do additional study to tease apart the mechanisms, but we certainly can say that individuals who live in highly disadvantaged contexts have more than their fair share of pollution exposure.
Great. Dr. Kine, I'm afraid we've run out of time. I want to thank you for joining us today and for your incredible work. We know there's much important things coming from you and your group and collaborations with us at UCI. So thank you so much.
Thank you so much, Dr. Grill. It's been an absolute honor to be part of your conference.
Thank you. And so now we're going to take a quick message from one of our sponsors before we come back to wrap up with the last talk of the day.
Vivante is a special new type of senior living community. Dedicated to the highest quality. Created, owned, and managed by members of our local community. Where we celebrate our heroes. At Vivante, we're committed to the health, well-being, and happiness of all of our residents. Come be part of the best.
With United Healthcare Medicare Advantage plans, there's more to take advantage of. Get a checkup here and have a wellness visit come to you there.
Wow, it's too good, the secret is out.
Pick up your prescriptions here and get free delivery there.
Wow, like everywhere, so word of mouth and wow.
Stay active with free home workout videos here and a free gym membership there.
Wow.
See a doctor in the morning here, or talk to a nurse in the moonlight there. That's more to take advantage of. Here, there, and everywhere. United Healthcare Medicare Advantage plans. Take advantage everywhere.
Wow.
Okay, so our last talk of the day will come from my friend and colleague, Dr. Greg Jica. Dr. Jica is a professor of neurology at the Sanders Brown Center on Aging at the University of Kentucky. He is the holder of the Robert T. and Niles Y. McCowan Endowed Chair in Alzheimer's Research at UK. Greg wears a number of extremely important hats at UK, running their clinical core in their Alzheimer's Disease Research Center, running their clinical trials program, He is a very prominent, important member of our field, an excellent investigator funded on numerous grants to do clinical trials, like what he will talk about in his presentation. I've had the distinct pleasure of knowing Greg for most of my career. We both serve on the internal ethics committees for the ADCS and the ACTC clinical trial networks. He's a pleasure to work with and a true leader in the field, as I'm sure you will agree after you watch this outstanding presentation. Enjoy.
Hi, I'm Dr. Greg Jica from the University of Kentucky, and I'm delighted to be asked to speak to you today on advancements in Alzheimer's disease treatment studies. I spend the majority of my time taking care of patients with Alzheimer's and related dementias, and a good portion of that is offering the opportunities to help us advance research and to gain access for many of our patients to clinical experimental therapeutics that they might not have access to otherwise. The same holds true at UCI. And so today we're going to take a whirlwind tour of these medications and of many of the advances in Alzheimer's disease treatment studies. We're not going to cover everything. And in some respects, there's going to be a lot of data presented to you, but I want to give you the big picture. No one left behind. So with that, my disclosures, none relevant other than I'll be discussing the unapproved use of many experimental medications today. And I'd like to thank you all for the support that I've received from the NIH, your tax dollars at work. So many people say we don't understand what causes Alzheimer's disease and what is actually happening in the brain. So how could we possibly develop medicines for it? And I'm here today to tell you that nothing could be further from the truth. We understand this process intricately. It begins as one is normal with the slow buildup of amyloid plaques in the brain. These kill nerve cells from the outside in. And this is rapidly followed by the development of abnormalities in another protein called tau, which leads to neurofibrillary tangles, destroying nerve cells from the inside out. As nerve cells begin to die, the brain begins to malfunction. We eventually start to see shrinkage of the brain from the nerve cell loss, eventually leading to memory loss and memory problems, and then further into functional decline, the stage that we've all recognized historically as representing Alzheimer's disease. So let's start with amyloid plaques in our search to leave no one behind. We've been working on amyloid for many, many years, and many of you may not be aware that the first Alzheimer's disease vaccine was developed in the last century. I feel old saying that, but it was true. And this was tested in humans. The initial data in mice that you can see in the upper right-hand corner showed a substantial reduction in amyloid plaques if treated with this vaccine. But in humans, it caused some difficulties. The trial had to be stopped, and we went back to the drawing board. Now, in addition to traditional vaccines, we've been working over the last several decades on passive immunization, and we'll talk about that later, trying to remove amyloid plaques from the brain with antibody infusions. We still are working on balancing the risks and benefits of such therapy. And it's become quite clear, we learn from every study, whether it's successful or not, some piece of information that helps us move forward as to whether or not the different types of amyloid that the antibody and or vaccine attacks or addresses may really matter. And we've also learned that we need to move to these earlier stages of disease, far before the time when one develops memory and thinking problems, in order for these medications that attack amyloid to be maximally effective. Again, I'd like to reassure you that safer vaccines are on the way and really are a goal of the field. So what are we doing with active vaccines? Well, we all saw that in just a little over a year and a half, we were able to bring COVID vaccines to bear, and we're trying to do the same with Alzheimer's disease. There have been dozens of active vaccines that have been developed. These are ongoing in human clinical trials currently. We have developed much, much safer vaccines that can move forward. We're working now on trying to make them strong enough to generate sufficient antibody responses in the aged population to be effective. So this is a story that I can't answer right now, but it is ongoing as one of our major efforts in the field. With that, over the past several decades, we've been focusing on passive immunization. Passive immunization is a different kind of vaccination. It is giving folks antibodies out of a vial. So instead of vaccinating you and allowing your own immune system to generate antibodies, which we may not be able to control, instead actually giving you antibodies that fight the disease itself. And this was popularized with our ex-President Trump. When he came down with COVID, he was rapidly whisked away to a military hospital and given remdesivir. Remdesivir is passive immunization, passive vaccination in a bottle. And these antibodies prevented him from getting significantly sick and or winding up in the intensive care unit. So this is a historic year because the first disease-modifying agent, aducanumab, has been conditionally approved by the FDA. And that means that we are getting close. The barrier has been broken. The aducanumab study was based on an antibody that removes amyloid from the brain. And two studies, the ENGAGE and EMERGE, tested aducanumab in over 16,000 mild AD patients. And the FDA had to concur that this removes amyloid plaques from the brain. There's still a question as to what the clinical benefit is, and there is some risk associated with these medicines. And we need to be careful with that, because the last thing in the world we want to do as physicians and scientists is to hurt anybody while we're trying to help them. Several other antibodies have moved forward as well with breakthrough designations by the FDA. And it's a race amongst the companies that are moving these medications forward, which one will receive the first full FDA approval. One has to ask, is these different antibodies are jockeying in position, are they all the same medicines? Well, they all remove amyloid from the brain, but I do think that the best way to understand this is to think about how you might catch a rattlesnake. Well, you could just reach down and grab it right in the middle of its body. It's a bigger target, but you run the risk of getting bit. You could grab it by the tail, staying away from the head, but again, that snake may turn around and bite you. Or, as most snake experts agree, the best way to catch a snake is by grabbing it on the neck right behind the head. This is harder to do, but you can successfully catch the snake without getting bit. And that's what we're trying to do with these medicines, because each of these antibodies catch amyloid in different ways, like catching a rattlesnake in different ways. They may catch just the amyloid monomers that we're all making in our brain. These are single pieces of amyloid. They may catch the early aggregation, what we call soluble oligomers, which are quite toxic to the brain and nerve cell function, or they may attack the plaque itself. What we do understand from these studies is that the more effective they are in removing plaques, the greater the risk of side effects are. And so trying to find that balance is critical for us as we move forward. Of course, one of the other pieces in our understanding is that the buildup of amyloid plaques occurs 10 to 20 years before one develops memory problems, providing a window of opportunity where we may be able to give these medicines and prevent Alzheimer's disease. And that is a major focus of research today. We do know that 1 out of every 3 persons over the age of 65 has this early buildup of amyloid plaques, and we do not want them to go on to develop Alzheimer's. Now, not all may, but this does put you in a high-risk group for the development of Alzheimer's, and we don't have to wait until your autopsy to tell whether or not you're one of those people. We can do this with PET scans and spinal fluid today. And so 2 of the major efforts funded by the NIH that UCI is part of, as with UK, include the A4 study. This is a study that has been ongoing for many years and uses an antibody that removes the monomers, the single amyloid proteins, to prevent further plaque buildup. We should see results in the next 2 years. The trial enrolled people who were still cognitively normal, but had that early buildup of amyloid in their brain. And currently, the AHEAD study is enrolling now. I urge you to get involved with this. If you're over the age of 60 with a family history and or 65 without a family history, this is designed to screen you using a PET scan to see if you have that early buildup of amyloid in your brain. And if you do, to give you a potential medication that might remove that. We think that when dealing with Alzheimer's disease, an ounce of prevention is worth a pound of cure, and we need to get to folks early. So please, UCI is active in this study. If you have a family risk or if you're concerned about your own risk for Alzheimer's, get involved. We need everyone to join in, no one left behind. So moving on to tau and tangles, this is very important because we know that we may all harbor a few tangles in our brain, but they stay relatively isolated unless those amyloid plaques release them, in which case they spread from nerve cell to nerve cell. And this was quite well established in 2012, independent research from 2 laboratories demonstrating that tau could be spread in that fashion. And we've developed PET scanning as well in living human beings that shows us whether or not you're starting to build up tau and neurofibrillary tangles. And looking at this again, as I said previously, we know that amyloid in beige here does not lead directly to dementia, but instead runs through tau changes and tau levels that are ultimately responsible for dementia. So if we may have missed the boat with cleaning the amyloid out of your brain, there's still hope that we can stop this tau propagation. And that's been studied for many years. So in 2007, this is an animal study in Alzheimer's, that is an animal form of Alzheimer's disease. We can see the Alzheimer's pathology in an untreated mouse, and we can virtually eliminate this with antibodies against tau. So it works in Alzheimer's disease. And in 2007, there was a worldwide party of mice across the globe dancing that they no longer had to fear Alzheimer's. But we're not there yet with humans because the disease in humans may be a little bit different, but nonetheless, we're intrigued by this. And currently there are 282 clinical trials of tau on clinicaltrials.gov. That's a website where you can find virtually everything in the United States that is a legitimate clinical trial. 71 of these are recruiting currently. With that being said, we've had some early troubles in this area. There have been several early phase 2 studies that have failed. The TANGO, the AWARE, and the AC Immune studies, all antibodies that recognize tau or the snake in the same place. And so is this a question that we need different tau antibodies? Is where the field exists currently. Can we catch that snake behind the neck where we don't get bit and we effectively neutralize it? That is our goal, and this field holds high promise in that regard. We know once the plaques and tangles begin, the brain begins to malfunction. And part of that malfunction is metabolic dysfunction. And so we've studied this over the last several decades. We know that metabolic processes like glucose regulation, sugar regulation, diabetes may predispose to Alzheimer's disease, leading to insulin resistance in the brain and a cascade that increases amyloid deposition and contributes to tau pathology. The same holds true for high cholesterol. And so metabolic function is clearly something that is on our target and radar screen. And so there are new trials coming out every day with new classes of medications to treat metabolic dysfunction. EVOKE is a trial that is just beginning worldwide, looking at agents for type 2 diabetes that correct metabolic dysfunction through the use of what are called GLP-1 agonists that increase insulin production. And in the animal models, these actually show that they prevent the cell death associated with the buildup of amyloid in the brain. We know that they have prominent effects on cardiovascular risk reduction, and this is a double plus. So we look forward to the results from these studies. And very important, if you have diabetes or high cholesterol, get those under control with your doctor. Do not carry that risk for future dementia. Once the brain malfunctions and we have plaques and tangles, the brain starts to shrink. This is because of nerve cell death. And what can we do at this stage of the disease? No one left behind. Well, excuse me, we know that those structural changes, the brain shrinkage, precedes memory loss by as much as 5 years, specifically in the memory areas of the brain. And we know that these memory areas actually can grow new nerve cells. So that gives us the potential to look at this stage of disease as a viable target. Brain atrophy as a whole may actually be worsened more by hardening of the arteries than by Alzheimer's disease atrophy. So this is a study from my lab at the University of Kentucky showing that 10% of the shrinkage of the brain is contributed to by simply getting older. 20%, just under that, may be related to Alzheimer's disease, but as much as 40% of the shrinkage of the brain may be due to hardening of the arteries, what we call small vessel ischemic change. On MRI, they appear as white matter hyperintensities. This may be another target for us. And using data from the Alzheimer's Disease Neuroimaging Initiative, this is a nationwide study that UCI is very involved with, trying to understand the progression from normal to dementia, that suggests that in terms of these white matter hyperintensities, this hardening of the arteries, that 55% of folks will progress inexorably, as we see with Alzheimer's disease, but 1 out of 5 may remain stable, and 1 out of 4 may actually heal and improve. And understanding why 1 out of every 4 people can heal is important, because we'd like 100% of people to heal from cerebrovascular disease, hardening of the arteries. And the first clue from this really came from the Sprint Mind study. This is a national study with thousands of men and women enrolled in it that looked at whether or not we could aggressively control blood pressure to prevent dementia. And these results came out just about 2 years ago now, targeting blood pressure. The American Heart Association says keep your systolic blood pressure, that's the top number, under 130. But in this study, the experimental group, we strove to keep it below 120. That's about the level where most teenagers exist. thinking teenagers are so darn healthy. If we could make the elderly population put their blood pressure in the same range, we may have success. And that's exactly what this study showed, that early memory loss, mild cognitive impairment, and or dementia could potentially be reduced. We could eliminate many, many cases by simply lowering those blood pressure parameters in folks. And that begins to shed some light on this area of metabolic dysfunction. As the brain progresses to lose nerve cells, we're still not leaving you behind. We're focused on those with memory loss and early mild cognitive impairment. We know that the average person over the age of 65 has a 1 to 2% risk per year of developing Alzheimer's disease. That risk increases dramatically if you've already had a memory and thinking problem, tenfold or more. And so identifying those with early memory problems is really a goal of the field. And we're not giving up hope that we can help these folks at this stage. So one of the major initiatives that's ongoing right now is called the EXERT trial. It is a national trial of exercise to help people with mild cognitive impairment, early memory problems, not go on to develop the functional difficulties and problems of full-scale dementia. We know from prior studies that exercise as little as 30 minutes 3 days a week. That could be just brisk walking in your neighborhood. It could be swimming, cycling, whatever you like to do, is enough to reduce your dementia risk by 60%. And why is this? Well, from the animal studies, we've begun to understand that new nerve cells grow in the memory areas of the brain, what we call the hippocampus, and that the growth and birth of those new nerve cells is highly dependent on BDNF, brain-derived neurotrophic factor, and that this is increased whenever you exercise. It's like miracle grow for your brain. So we can see in the bottom panel, in the middle, green nerve cells, those are existing nerve cells, and those you see in orange in an unexercising animal, that means a sedentary lifestyle animal, we still see them growing new nerve cells. But look at the explosion of new nerve cells when we allow the mice to exercise, give them a wheel in their cage and little tubes to run around in. And if we look at the nerve cells themselves, look how much healthier they are in the animals that experienced exercise. The best information we have is that the exact same holds true for humans. We know after about 20 minutes of exercise, the BDNF levels circulating in your bloodstream and getting to your brain increase dramatically. So it's not just about drugs. And a lot of what I've talked about today is about medications. It's also about lifestyle. And it may not just be physical exercise. So one of the major discoveries over the past several years has come from the finger study in Finland that actually looked at a multi-component lifestyle intervention. So let's not just fix your exercise and get you up and running, but let's cognitively stimulate you with brain exercises. Let's take care of your medical problems like your cholesterol and diabetes. Let's start you on a healthy diet. And that study showed that we could prevent cognitive decline in folks that were undergoing this intervention. And that's led to what we call the US Pointer Study, a major national effort funded by the Alzheimer's Association, in addition to the NIH, that is looking at this combination. Let's help people to exercise, give them the tools to have a healthy diet, cognitively stimulate them, exercise that brain, and monitor your heart health risk factors. And that study is ongoing. I encourage you to follow that and to adapt these healthy lifestyle interventions to reduce your risk. At the end of the day, we don't have a cure. And so we know that everyone coming down with Alzheimer's disease or a dementia is going to progress. Well, what are we doing about that? Well, we're doing a wealth in the area of Alzheimer's disease, but I'd like to kind of change gears a little bit here because this has bearing on some major work coming out of UCI. So this is a case report of a patient followed at UK longitudinally for over 20 years from the age of 77 and 91, did quite well on the red line, as you can see. And then in 92, the memory began to drop, and that persisted with a diagnosis of mild cognitive impairment and eventually full-blown Alzheimer's by 97. We were surprised when she eventually passed away and her brain came to autopsy that she did not have Alzheimer's disease. Instead, she had something that we called at the time hippocampal sclerosis. This is a loss of nerve cells not caused by plaque tangles, but instead caused by a protein called TDP-43. And this has been termed late limbic associated or late onset age-related TDP-43 encephalopathy. Why? Because this occurs in the memory and limbic areas. Age is the number one risk factor. TDP-43 is the protein that aggregates and it causes nerve cell loss and eventual dementia. And we've been able to discover that TDP-43 linked to many degenerative diseases such as FTD in a much younger population can cause neurodegeneration, nerve cell death through a variety of pathways. And I always hate these busy diagrams. We're not going to get into this, but let me reassure you, we understand how this protein is playing a role in nerve cell death. We've also been able to look at the genetics of folks with late. And what we find is that there are some genetic risk factors, as we know for all the diseases, Alzheimer's, Lewy body disease, frontotemporal dementia, and one in particular, ABCC9, is of interest to us. We know that this gene encodes for a channel, a potassium channel, and that this potassium channel helps regulate nerve cell health. Originally looked at in cardiac disease, there have been medications that have been developed to modulate this channel. And unfortunately, they're not available in the United States, but are approved in many other countries. We know that people treated with nicorandil actually have a third of the risk of death from cardiovascular or cerebrovascular disease, but we think the same may hold true for their impact on late in terms of brain cell death. And the folks here at UCI, Claudia Cowis with her 90+ study, have really been leaders in this field, demonstrating that late is a major problem in Alzheimer's disease mimic. And we are moving forward at UK with an NIH grant and FDA backing to bring nicorandil into this country. We are beginning the first ever clinical trial for late. No one will be left behind. At the end of the day, however, until we have a cure, we're eventually left with end-stage Alzheimer's disease, a brain destroyed. We can't fix a brain that's so damaged. Reversing or rebuilding the brain may be many years in the future, centuries, if at all possible. But as I hope I've expressed to you, we may be able to slow or stop the disease at an earlier stage, and through the development of new vaccines, whether those are active or passive, we may be able to prevent AD, hopefully within the next decade. But no one left behind. What do we do about those that are in the end stage of dementia? Well, another case report. This is Joan, 82 years old, in the end stage of dementia. She's bedbound and does not speak. As she lays in bed, she frequently moans and grimaces as if she's in pain. All we can really do is keep her comfortable and be there. It's impossible to know what she's feeling. We can give her standard hospice medicines, and she is on hospice, morphine and Valium to make her more comfortable, but then she's even less aware and doesn't recognize her son when he comes to sit with her, hold her hand, kiss her on the forehead. When she's awake, she appreciates, and it's the only time in her day that you actually see her smile. There has to be more that we can do, and no one will be left behind. Well, discoveries have been made in terms of cannabinoids. Cannabidiol and THC may actually help regulate some of these pathways that are involved in the late stage Alzheimer process, and these medicines may be used to provide additional comfort. Again, I'm not going to get into the biochemical details here, but I do want you to know that NIH has put forward and funded a clinical trial from the Alzheimer's Clinical Trial Consortium. That is a nationwide consortium that UCI and UK are part of, to actually look at this to see if we can reduce dependence on morphine and Valium-like agents and provide comfort in those final days of the lives of our patients with Alzheimer's disease. We look forward to this study. Again, no one will be left behind. So when it comes to Alzheimer's disease and research, I want you to really appreciate the profound progress that we've made, the hope that is on the horizon for the near discovery of agents that may slow or stop the disease, that may help treat the disease, that may help provide comfort with the disease. Why might you consider participating yourself and or your loved one? Well, one would be to go beyond standard treatments rather than just rolling over and giving up. And many of my patients express that to me. What else can I do, Dr. Jaika? What else can I do to fight this disease? And I can arm them. I can tell them exactly what their options are. Never give up. Fight. The most important reason is not just for us. It's for our children and our children's children, for the generations to come. My hope and prayer is that they grow up in a world where we no longer know Alzheimer's disease or the ravages of the other dementias. Thank you so much for your attention today, and I'm available for questions. I appreciate your attention, and really, again, thank you to the sponsors of this program.
Okay, we are now joined live by Dr. Greg Jaika. Greg, thank you for your talk. Thank you for being here. I wholeheartedly endorse and love your message of no one left behind. We're grateful to you.
Well, thank you, Josh. Thank you for the introduction as well. And hello, California. As I was telling Dr. Grill, my son is at Chapman right around the corner from you all. I have a vested interest in the health and well-being of California, and I was delighted to present at least what I could to you all today, although just a snippet of the exciting things going on in the field.
It's great. It was really a fantastic talk. And your first question comes from Dr. Liz Head, and I had a similar one for you, and that is, what do you think the impact of the FDA approval of aducanumab, Aduhelm, will be on our ability to do clinical trials for Alzheimer's disease?
So this is an important consideration, and certainly something that is created ground tremors, but not a true quake in the field. And I don't think we're going to experience that. When we really look at where the data has come from for the study, there has really been a pushback to say that folks engaged in the Engage and Emerge trials that led to that approval were of a particular subset. They needed to be early in the disease course. We need to be able to do MRI scans. And so while initially we were looking at, maybe half of the 6.2 million people in the United States being eligible for aducanumab, just yesterday, actually, in JAMA Neurology, a paper came out looking at Medicare claims database that shows that, no, it's actually only really a small percentage, about 8%, 8 to 9% of folks are actually going to be eligible. That leaves people behind. That leaves 91% of people behind. And we can't have that, as I said in my talk, the paraphrase, no one left behind. So I do think that we're going to have some, but people really need to realize that this was a conditional approval. It requires an additional study to be done. As such, in essence, this is an experimental medicine, no better or worse than anything else that we have for experimental medicines. It's just been approved for your doctor to potentially prescribe this to you. So I actually don't see much of an impact at all. And it may be right for some individuals. For the individuals it's right for, that's appropriate. And for those that is not the right choice for, there are so many other options that are going to drive the field forward. I think we're going to do this together, Josh.
Yeah, well said. And would people with Down syndrome and Alzheimer's disease be among those for whom this drug could be right for them?
That's a great question. And currently, as the Engage and Emerge trials were done, and currently as folks are looking at interpretations of who might be eligible or not, those with other neurologic diseases or developmental disabilities do not seem to fit into that approval, largely because we don't yet understand not so much the efficacy, but we don't understand the safety concerns and risks. And I work with an adult Downs population. These folks are precious. And the last thing in the world that we want to do for anyone who's added in experimental medicine as an adjunct to their clinical care is to create any harm or safety issues. So always protect. As I always say, we don't have research participants. We have patients that take experimental medicines. People think that that is just semantics, but I say, no. If you say research participants, the research comes first. If you say patients that take experimental medications, we place it in the appropriate context. It's the person that comes first. That's who we're caring for. That's who we're trying to allow to go above and beyond what you can get at your local pharmacy, and to be part of the discovery, to contribute to helping the millions today and the many, many millions tomorrow, our children, our grandchildren. We're doing this for everyone.
Now, you mentioned safety, and of course, aducanumab and other anti-amyloid therapies have, like any drug, safety signals that we should consider. One of our audience members asked you to elaborate a little bit about thinking about safety, perhaps especially in the context of prevention and preclinical AD trials.
So very, very important. And again, Josh, the analogy to rattlesnakes was not meant to imply that California is full of rattlesnakes. It actually came from where I grew up in Connecticut, where there are many rattlesnakes. And I actually lived on a farm in an area where there was a high rattlesnake population. As a kid, I used to go out and catch rattlesnakes. And that analogy was meant not just, if we're trying to catch the snake, there are lots of different ways to catch it, but you want to catch it in a safe way. That's incredibly important. And so when we look at these different studies, we really have to look at the individual person. what are their potential risks? Is this going to be unsafe for them individually, but it might be much safer for somebody else? And pick and choose. And this is where you really need to work with your doctors, with study teams at UCI to figure out what's best for you, always with safety first. I always say, you know, I've caught many rattlesnakes in my life. You catch them right behind the head, as close as possible on the neck, and you will never get bit. And that's what we need to do with our clinical studies and with our drugs. Pick the rattler that you can catch right behind the neck, so you have as little chance as possible as experiencing any side effect. And remember, you know, we all go to the pharmacy to pick up the medications our doctor prescribes us. We read that package label. It says, oh my God, it can do this, that, and the other thing. But remember, keep these risks in mind. We want open disclosure. Most people taking pharmaceutical medications do not experience horrific side effects, and most people in clinical trials do not experience horrific side effects either, or the trial would be shut down by the FDA, which approves clinical trials the same way as it approves the medications we receive from our local pharmacies.
The lawyers are buzzing in my ear telling me to tell the audience we are not recommending anyone try to catch rattlesnakes today or any other day.
Yes, and I'll second that. Don't try this at home.
Unfortunately, I only have time for one more question. There were a few in the chat. I want to just get to one of them. Can you address whether low blood pressure could have a role in Alzheimer's disease?
Yeah, and certainly we know that the relationship of blood pressure with dementia as a whole is paradoxical. In midlife, we really do want very low blood pressures, but there is some evidence that as we age, lowering the blood pressure too much could be harmful. With that being said, I presented the SPRINT study, which actually was a proof of concept of this in an aged population, still relatively young in overall in their low 70s for a group as a whole. But it showed there that the blood pressure that a teenager has that is healthy for a teenager is also healthy when you're 70. And I think of 70 as a teenager, you know, in my clinic anyway. And so I think, you know, what's applicable for good health should not have age restrictions on it. However, if lowering the blood pressure creates symptoms, that is problematic. And clearly that would tell us on an individual basis, we cannot go that low with this individual person. That actually was incorporated in the SPRINT trial study for those that saw that. If patients became symptomatic, they were not pushed to the limits of their randomization group.
Great. Greg, outstanding talk. Thank you so much for being here. Thank you for everything you do for the field. We're grateful to you. Thanks for joining us.
Thank you so much for having me on today, Josh, and thank you, California. You guys are a powerful force in our search for new and better medicines that are one day going to lead to the cure for Alzheimer's and all related disorders. So enjoy the rest of your conference. It's really been wonderful.
Thank you. And with that, it actually brings to a close our 2021 Southern California Alzheimer's Disease Research Conference. I hope that you have enjoyed this remarkable slate of speakers who I think deserve a lot of gratitude for just a fantastic set of interesting and diverse topics. I want to also acknowledge the tremendous work that went into planning this event, especially Mark Odom, Tuni Chatterjee, Aaron Gutierrez, Melissa Klabe, and Veronica Majori from Alzheimer's Orange County, and Megan Whiprock and Kirsten Klein from here at UCI Mind. We hope that we will see you all again in the very near future. And when we do, please congratulate these folks who worked many, many hours to put on this really excellent platform. I also want to thank my colleagues here at UCI Media. I am literally standing in front of a green screen in the studio. Will Alvarez and Kyle Good made it possible for us to do this. They've been showing great hospitality to me here in this studio and worked hard themselves in helping with editing the videos and producing a seamless technological conference today. We hope that you've enjoyed yourself today. Keep your eye on the UCI Mind and Alzheimer's Orange County websites for new opportunities, educational programs, and research opportunities. Until we meet again, stay safe, and we'll see you soon.